All right, so welcome to QC Bio Workshop 6, February 2021, DNA methylation BS-Seq analysis, day 2. So we're going to continue where we left off, and I think it's better for me to kind of get the basics of the analysis pipeline, the core primary analysis, really well and done, and then some of the downstream stuff and so on. That's easier for you to kind of pick up on your own, and it's so much variability depending on what your project is and so on that, and if you don't have the fundamentals down, it's sort of not as useful to rush to that. So today we're going to cover the alignment and quantification, and that's probably going to take the whole time, and that'll give us a solid foundation for what comes next and also your own work. Okay, so the slides you should have an email, and I will start screen sharing here. And we will have the slides. And we have 2 terminal windows to add. In the Firefox. That should be good. Okay. All right, so the terminal windows we don't need right now, so I'll push those aside. Why don't we make, yeah, okay. All right, so here is where we're at, right? So this is the generic pipeline. And I should pop out my chat window as well. There we go. And we've covered the initial QC with a fast QC. We did some trimming with CreditApp, and I left you to look at the second QC post-trimming yourself. So hopefully you're able to do that. Since people are reporting Hoffman is a little slow, maybe get on Hoffman, so log in, and then ask for your work, your interactive node, and we'll try with the same research requirements that we asked for the last time. And if that's turned out to be real trouble, we're going to have to lower them. Okay, so start that, and we'll leave that for a few minutes and come back, and then maybe it'll be ready once we're there. Oh, okay, it didn't take me long with those requirements. Make sure you use the little I here, which is for interactive, which kind of gives you a boost in the scheduler rankings. It knows that a human's waiting on that, and it gives you a boost. And once you get on, start copying the files we'll need for today. So let's just make this little shell variable for your personal, this is your personal scratch space. Okay, and then switch to my scratch space. Of course, for me they're the same, but okay. And there's my originals, and you need this day 2 start state directory tree, and so copy preserving permissions recursively with progress information, and you overwrite safety, but you're not going to have any conflicts there. And so copy this day 2 start state directory into your personal scratch space and call the copy day 2. Okay, and let that go, and that'll start copying files, and we can let that work, and we'll come back to it, and hopefully you'll all be synced up once we're ready for it. Okay, so I've given you even the reads again in this day 2 folder. I just decided to make sure everybody's starting from the same state again. And so it's got the reads and everything. It's got a copy of everything we did in day 1 and including the reads. And it just gives us all a known state to start from in case you've been playing with the day 1 directory on your own. In real situations, of course, you wouldn't like gratuitously duplicate your large sequence files like that and so on. But anyway. And this UNIX stuff where we kind of push together all these single letter flags is very common, at least with traditional style UNIX utilities. Of course, CP is one of the oldest ones. It's probably 40 years old at this point, 50 years old maybe. You can take the flags that are single letters long and mush them into a single argument as you please. So this is equivalent to writing out this. And in fact, that's why the long options that are words, multi-characters, start with double dash, because otherwise there would be a conflict between interpreting one of these long guys as single letter P, R, E, S, E, R, V, E, all conjoined together, a union of many single flags versus a single long option. And so that's why you need the double dash to distinguish whether you're talking about a merge of single letter options or a beginning of a long single option. So this PRVI is also equivalent to this four long ones spelled out, but of course it's much easier to type this. Okay, yeah. So hopefully you guys all did rerun FastQC and you made your own script to record, make your kind of your working log of what you did, and then cleaned up in the same style that we had going. So your script probably looks something like this then. So the stage 3 to 4 script that runs FastQC after cut it out, just some start and stop, and we call FastQC, and now we pointed at the stage 3 post, the stage 3 directory has the post cut adapt FastQs, and so that's what we feed into FastQC, and then we run it, and it's so fast, and these are small files, you can just run it directly in an interactive session. We don't have to use the job submission queue. There it is, it ran. And now that it made the outputs, we want to isolate them into their own stage 4 directory. This is the FastQC after cut adapt stage. And so we move those files from the stage 3 place where our usual setup is to generate the next stage's files in the previous stage's place. And then once we know it's all fine, then we move the new stuff into its own new stage directory. Okay, and then you would need to, so here's what the kind of the setup looks like in the working space after that's done. We've got our scripts that are recording how you want from stage to stage. We've got stage 1 base FastQs we started with. We've got the stage 2 FastQC outputs on stage 1. Then we got cut adapt, which took the stage 1 FastQs and trimmed out to nucleotides 11 to 74. That was our decision. And so we have new FastQs in stage 3, and then in stage 4, we've got the FastQC run on stage 3. And then you're going to need to download those to your local machine. So that's just similar commands to what we did before, or however you want to do it, whatever your file transfer mechanism is, and the GUI or whatever command line, you unzip the unzips and you open the HTMLs in your web browser and you can view them in your local machine. Okay, and so hopefully everyone's got that done. And okay, my copy of the files to my working session at Hoffman is complete. Hopefully yours is progressing or done. And let's see. Here is the place where I have these. I think I have these. No, where did I put them? No, we'll just get them fresh. Okay, so here's my working machine, my local machine, and I'm on my desktop. I just made a day 2 directory in my desktop. This is where I'll do stuff on my local machine. And for me, I need to copy those files, so conveniently, command is almost there, except it's now called day 2. So there we grab them, and there we're going to. This time I copied the whole directory recursively rather than ask for the files individually. So if you look, it made the SCP-R as a recursive copy. So now I've got a copy of the stage 4 directory, and I'll do the little unzip magic. And now, yes, if you've copied the files for today, you'll notice that you've probably got some permissions denied errors because I haven't made the scripts for the BS-Bolt stuff readable to you guys yet. So that's what that looks like when that happens, and we'll grab them as we get there. So let me use Firefox. I've got Firefox in the screen share right now, and so we'll open up. So here's our new FastQCs, which hopefully you got a chance to look at. And if you look now, the Q-scores are more flat across the thing, and the base composition is more stable across the end. From one read to the other to the end. And there aren't any more adapters anymore because the reads that are so short to read into adapters, it was about 5% at the worst at the end of tail end of some of these guys. Those are gone now because we've cut off before that point. So that's one of them. You know, there's 6. And if you look through all of these, they were all looking pretty good. If anything, we trimmed too much, right? We had that discussion yesterday where we've kind of taken a fairly significant hit to coverage, and coverage is sort of precious here. So maybe in a real analysis, we would really be this aggressive with the trimming. But this was easy to do, and it takes care of the problem for sure. Okay, so that's that. So we're there now. Okay, so let me ask. I know that one of you guys has gotten the copy to basically be done. How about other people? How many other people got a session on Hoffman and have finished the? Where are you at? Did you finish? Did you get a session? Did you finish your copy? Your copy is still in progress. What's the status? Okay, 2 people copied. 3 people copied. 4. Sounds good. All right, so now we are going to talk about BS-Bolt. And that is a nice recent package based on Python out of the Pellegrini lab that really wraps up a lot of the analysis that you need to do for the primary analysis of a BS-Seq experiment. It can handle WGBS and RRBS, but we're not going to really talk about the way you handle RRBS. You basically have to kind of tell it what are the regions you've targeted. And we're just going to do the basic WGBS. Okay, so this is the page for it. There is a bioRxiv paper. I don't think it's published yet in a peer-reviewed journal. I'm sure that'll come. This is the online documentation and so on. There's a link to probably a GitHub page that's got the source code too, if you care. And we're going to have to install it. But we've already gotten practice last time with Cutadapt and so on installing Python stuff. So this really isn't too bad. Okay, so here I am in my Hoffman window. Maybe I'll make that a little narrower too. I don't really know what you guys see. If you guys see the exact same window layout that I see, and so when I have overlapping windows, you see overlapping windows. Or, anyway, so we're going to be using Python, so we got to do the module activation business again. So we're going to use Python 370 like we did last time. So let's activate that in our Hoffman environment, and let's check if it's already installed. So pip3 list tells us what Python packages are installed, and yes, I have BS-Bolt 144 installed. Now, one thing that's a little confusing is at some point in BS-Bolt's history, it used to be spelled BS-Bolt, capital B, capital S, capital B. And some of you may have that already installed as an old version and so on, and then it's kind of confusing. So in that case, if you've already got the old version installed, you probably want to uninstall it first. And so the way you do that is pip3 uninstall BS-Bolt with the capitalization like that. I'm not going to run that because I don't have it. That's a Control-C to cancel the line there. And then do the install for the current version. So if you don't have it, or once you've done the uninstall of the old version, then you can do the install. So then do a pip3, put it in your personal use, tilde, home directory tilde /.local. That's the # user option to pip3 install, and it's now called BS-Bolt all lowercase. Okay, I can run it. It's not going to harm anything. I've already got it installed, and basically you'll see that pip3 says, okay, you've asked for it, and it's already there. Requirement already satisfied, and then all the dependencies are also already satisfied. So it's nothing to do. All right, so give me a ping in the chat if you successfully installed BS-Bolt, current version 144. Okay, and let's one person installed, and we've got the path business. Okay, so we add that .local bin directory there. So actually, we've got Cutadapt at this point too. We've got everything that's in your local bin, whatever it is. I have a few things, not that many. Actually, BS-Bolt these days, because they have that history of calling it with the B, capital S, capital B, actually installs 2 scripts to launch it, which are both the same. these days. So you can invoke it with BS-Bold, which is the old name, but I think I will try to use the new name and just stick to that. As I say, those two launcher scripts for it are exactly the same. They launch exactly the same thing. So, and it's just due to history why they have them both. I think they were forced to go to the lowercase because that's the convention for some of the Python environments, like Bioconda or Conda or whatever. Okay, so yeah, so BS-Bold has a whole bunch of little subprograms in it, modules, they call them subcommands, whatever. And so the main ones we're going to run today are index to get our reference genome ready. Then we're going to align our FASTQs, our cut adapted FASTQs to the reference genome via the index. And then we will be doing the methylation pileup to get the statistics of observed and methylated and unmethylated read base calls sitting above each genomic cytosine site. Okay, those are the modules we're going to use today. And the way you get help on one of the modules is to just give the name of the module with no arguments. So the first module we're going to need to run is index to prepare our reference genome for use by BS-Bold. Like most aligners, BS-Bold, which is internally based on BWA, needs to pre-compute some fast index structures on the reference genome to be able to rapidly align the reads to the reference. Okay, so the index module takes these options. Let's see here. You got to give it the input genome FASTA file. That, of course, totally makes sense. It's tolerant. You can give it a gzip one, and that can be a multi-FASTA file with all the entries for the genome in one file. I think that's the only way it really wants it. Then we give it the name of a directory that it will create, that it will populate with its index information, what it calls the database, the DB, or the index. Then these are some parameters you don't need to worry about. The defaults are just fine. There's some stuff related to RRBS, which aren't relevant for our example. And so this is very simple. The only thing we don't have right now is that we don't have a reference genome. So let me make sure your working directory is in your day 2 in your local scratch. So now your PWD is in your current working directory is in your copy of today's stuff. Okay, so you won't have these stage 4 to 5 and 5 to 6 scripts yet. The permissions weren't set for you. Then you can see that I didn't make them yet readable to you guys. That's why the CP couldn't get them. You can see this directory though, which is why you knew that their existence and why CP complained that it couldn't get at them. Okay, so we need the reference genome. We don't have one yet. Okay, so we got to go dig out the reference genome. Okay, so that comes from Ensembl. I do have a Firefox still around here, so let's go to Ensembl. All right. So my S key turns out to be dying on this laptop, as is my up arrow, and so often my S's just go missing. Okay. All right, so here's the Ensembl website. We are using Ensembl. This is still Ensembl 103. That hasn't changed. We just had a release, so that's going to be a couple months before they change. Here's human, GRCH38P13. That's where we're going. And then there's two basically important sides to this. Here is where you grab the sequences, the ACGTN sequences of the genome. That's here. There's also, we'll need some annotations. We'll need the genes and stuff because we're going to do some IGV visualization today. So that we will get from here, but we won't need that till later. All right, so we're going to click here, which is where the FASTA sequence is. And then this is really an FTP site. And recent versions of Chrome and Firefox have deleted FTP support directly in the browser. I'm not totally sure whether Ensembl now has an HTTP-based wrapper around their FTP site. So if you're running a bleeding edge Firefox or Chrome, you might want to, it'd be great if you could tell me whether you can still see this, which is probably going to be done by Ensembl dynamically translating their FTP site into reflecting it into their web browser using some module on their backend web server. So can everybody see this? Nobody's got some kind of can't go there, or I don't know what the error message is even with a modern Chrome or Firefox when you go to an FTP site and it doesn't have that support anymore. So can everybody see this place? So there is the primary assembly would be if you want the whole thing. That's got chromosome, it's got chromosomes, all the chromosomes, including mitochondria and XY. It's got a whole bunch of unplaced scaffolds too, but it doesn't have the alternate haplotypes. Right? At this point, we know enough about the human genome that we actually can assemble large kind of chunks of alternative haplotypes to the nominal primary assembly.
Excuse me, Sean, can I ask a question?
Yep.
Yeah, so on that site then, you've got 3 different types of files, right? One's DNA without the RM, one with RM, and one with SM. So can you talk a little bit about the difference between them? And
exactly. So they're basically all the same information. Actually, the RM has less information. The one with the most information is the SMs. The RM has the least, and the one on the top is I guess, in the middle. So as you know, genomes have a lot of repetitive sequence, right? We've got all these transposons, sign elements, line elements, and so on. The genome's full of them. And so for certain analyses, like blasting stuff into the genome or something, you at least want to know where those repetitive sequences are, and and they may be half the genome or what. In plants it could be higher than that, right? It could be crazy high. So it's useful to have the locations of those repetitive sequences marked. And so, for example, aligners that are in a mode where they sort of want to ignore the repetitive stuff can sort of know that with the marking where to sort of ignore sort of hits that might be just sort of non-useful. And so one way to do that, the kind of the most convenient way to do that, the genome primary sequence is basically ACGT. Well, there's about 5% in the human genome where we don't know right now, and it's currently represented in the genome by using ends as gaps, right? So that's just, an N is a position we don't know much about. We know it sort of exists, but we don't know what base goes there. So anyway, there's about 5% that are ends. But anyway, the primary sequence is ACGTs, and if you like, ends. But to indicate where the repetitive sequence is, one possibility, and which is often done, is to use lowercase letters. And to use the lowercase ACGT ends, and the lowercase n doesn't make much sense versus capital N, but the lowercase ACGT can be an indicator that is somebody's declaration of repetitive sequence, whereas the uppercase ACGT is the non-repetitive sequence. So the SM versions, which are soft masked, that's the SM, soft masked, SM, initial letters, that is with that style, where the repetitive sequences have been identified by putting the ACGTs there in lowercase. The RMs are hard masked, they are repeat masked, and the repetitive sequences have actually been replaced with ends. So that sequence is no longer informative. They don't delete it because that would shift all the coordinates around, right? So you just end it out, and that way it's not seen, but you've lost information by doing that. And of course, for our analyses, you wouldn't want to do that. If a read comes from a repetitive region, we want to align it there to the extent possible. The regular version that's neither RM or SM will be with all the sequence in uppercase, where the distinction just isn't considered. Okay, so that answers the question?
Yep, that's great, thanks.
Okay, so I usually get, yes?
For reference genome between gene code and Ensembl, can you comment on that?
So the primary sequence these days is, I think, moderated by GRC, which is the consortium. I don't think people disagree on that. So whatever GRC declares as, GRC H38 is the major version. There's also patch levels. Like we saw that sort of on the, if I go back, maybe it's too back. This is GRC H38.P13. It's the 13th minor revision to the H38 major revision. It's the 13th patch. Let me go forward again, so we're back over there. So that's, I don't think all the different places, NCBI, Ensembl, DD, the Japan database, everybody agrees on the primary sequence, these days, because that's managed by this GRC consortium. Where people sometimes vary a little bit is in the annotation, the genes, and so on. Maybe the non-coding annotations, like this. The gene builds are often have some secret sauce that each group likes to apply. And so sometimes there's variations in the gene annotations, and that's more where you get some variation. And certainly each group tends to have its own systematic identifier system for genes. So, not, humans, we like to use letters, ADH1 or something, is a gene name to a human, but systematically that's very hard, those gene symbols are often very hard to match up to particular gene models in particular annotation releases, which is why at the computation level, we often use the systematic identifiers, which usually look like horrible things where, it's like ENS 9 digits or 11 digits or whatever it is, right? Some sort of serial number that doesn't mean anything except to the machine, but it's at least a very precise way of identifying which model you're talking about in a particular annotation release. So yeah, it makes sense to use a source for the annotations that's going to give you, that's the richest source of information relative to the thing you study, right? So if you have other functional databases or something which are based off Ensembl, systematic accessions, maybe you should be using Ensembl stuff just because it meshes well with all the other, with the other data you're kind of interacting with and so on. But and certain organisms tend to have preferred sources and so on. And certain organisms like Arabidopsis, there's another consortium, TER, that sort of mediates Arabidopsis, and they sort of own the genes for that. Although I think also zebrafish has ZFin, although I think the sequence for zebrafish is now managed also by the GRC consortium. Anyway, it's an evolving situation and so on. But over time, usually sort of for especially the model organisms, some group becomes placed in charge, and at least of the assembly, if not the gene models, and then sort of people more converge as to what everything is. But especially for non-model organisms, it can be sort of chaos, right? It's not really managed by any of the central places. It's managed by the group that's most interested in doing it, or maybe which group did the de novo assembly of the genome. And anyway, it just varies a lot organism to organism. But the model organisms are very stable and very well known, and over time everyone kind of converges. Does that kind of help?
Thank you.
Okay. So yeah, the primary assembly ones they're all merged together. These all guys have the alternate haplotypes, which we don't care about. But we, of course, only need chromosome 21, and conveniently, they're all broken out here. So we will grab the, say, the softmax version. As I say, it's got the most information in it. The lowercase letters generally don't harm most things. Most things just ignore the uppercase, lowercase, including BS-Bold. So we've got the 21. We need the 21 here. Sorry. So I've grabbed the location for that. I've got the URL. It's in my clipboard, my GUI clipboard. I'm sitting back in my Hoffman2 personal scratch base, and I'm going to wget it and download a copy of it. And I think, did I do it a snazzy way here? So no, I didn't do it there. But here, I'll show you. We like to keep a log of what date we downloaded it, and how big the file is, and what the URL is, and so on. So here's some little magic to sort of keep track of it. Now we know what the file name is going to be. This is going to be the file name. So, crap. Looks like my copy and paste got the carriage return in there, and so the command activated before I was ready. All right, so I'll have to back up and show you. We're going to get the file, and the log file is going to have the wrong name because the command line got cut off before I was ready. And actually, that looks too big. It's scary big. Did I get the right one? Did I get the whole one? 21, should be this one. Yeah. Let's go check on that term. Is it done? Yeah, there we go. Okay. I guess that's how big it is. So this I tried to make this log file. Let me rename it to what it was supposed to be, and then I'll show you the command that was supposed to generate it. Okay. So that's the name it was supposed to get. It contains basically a copy of everything we saw scroll by as wget did its work. Okay, so this is the contents of our log. It's got the date. It's got the URL. So it's everything we would need to know to kind of state exactly what we got. The command to do it was this. That's what it should have been. It just the carriage return activated it before I was ready. So wget, the URL, the usual thing. But then I'm just going to redirect standard error to standard output. So both my standard error and standard output are going to the same pipe. And then T is a nice thing that takes standard input to standard output. It basically does nothing, except it also diverts a copy to as many other places as you like. So it diverts a copy of these combined standard out and standard error streams now into this file. So it's a way for us to both see it live on the screen as it happens and capture that into a file. So that's we've now documented our retrieval of this thing nicely. Okay, so yeah, so make sure you get that file and make sure it's on your Hoffman, because that's our reference genome, and we need to start feeding that to BS-Seq. Now let's take a look. You know, we haven't looked at FASTA files yet in this workshop, so let's go look and see what's inside one of these things. It's a gzip file, text file, no problem. All right, we know that less on Hoffman can handle gzip text files, so I'm just going to view it with less. And there it is. All right, so there's the, you know, the way FASTA files work is you've got that greater than sign at the top, which is the name of the sequence. And then in this case, the name is 22. One thing people vary about is whether they would call that CHR22 or 22. And in fact, you know, whether the C is capital, you know, it's capital C, lowercase HR22, lowercase CHR22, you know, whatever, maybe capital C, capital H, capital R22, maybe that's probably rare. But anyway, so people sometimes vary whether to put the CHR in front or not. At least the Ensembl style is to not put the CHRs there. And then usually sort of the words that come after that point are sort of optional. And a lot of programs, including BS-Seq, are just going to throw these away. And it's just going to keep the first word, you know, words being separated by white space. I think it's chromosome 21. There's a question whether it's 21 or 22. And it should be 21, because I think 21 is the smallest. 21 is the smallest autosome. Right? Yes, it is. Why? And then why is chromosome 21 smaller than 22? It's because when we decided on what the numbering was, we didn't know it precisely enough yet, and we kind of got it wrong. Such similar thing happens in many genomes. You know, ostensibly, I think the default, in the absence of all other considerations, the default is to number the chromosomes by descending length. And usually, if the centromere is known, you put the, you know, I guess you put the short arm on the left. You know, that gives you a way to decide strand. If the centromere is near the middle, who knows? But anyway, yeah, it's only a guideline. There's usually exceptions as things get more resolved as genome projects evolve. So we're, you know, BS-Seq's going to call this 20. Oh, what did I do? Did I download 22 and I need 21? Is that the problem? Yeah. Okay, that looks like the problem. Anyway, we'll get it right eventually. Okay, so here's 21. All right. Somewhere there's my wget line. All right, I can just change this 21, 21. All right. You notice the progress bar style changes when it knows it's kind of going not to your screen. W gets a little smart that way. All right, there we go. All right, so I got both now, but I think you only need 21. So we can put Firefox away. Yes, we were looking at 21 should be it, right? Maybe I should just delete this 22 so it's not confusing. Right, so goodbye. Okay, now we got 21. Okay, so there. Thanks for that. Otherwise, I would be more confused later on. Okay, so the beginning of this thing is obviously gappy. I'm going to hold down spacebar and let spacebar key repeat and see if we can get past this gap region. It looks like it's pretty long. You know, anyway, okay. How about we go to the end of the file? So if you type a greater than sign, usually shift period on your keyboard, that'll go to the end of the file. There we go. And well, that's gaps too. Okay, if you go backward a few pages though, B key, remember, is to go backwards a page. I'm gonna, oh, it doesn't take long, right? So the gap at the end gap at the end is pretty short, this guy. In fact, actually, you can see the telomeric repeats here at the end, right? So this stuff at the end, that looks like the telomeric repeats there. Anyway, that's what a FASTA file looks like. And you saw that because we are using the softmax version, you see lowercase ACGTs and you see uppercase AGTs, the lowercase being a repetitive sequence, and the human's a pretty repetitive genome. Lots of dispersed repeats. So there's lots of lowercase sequence all over the place. All right, so here I was documenting how I retrieved it, but we've done that at fancier right now, so I don't have to do it. Okay, just as an aside, if you ever just need to know the contents of a really large one that you can't just sort of skim like we just did, I have put in my personal scratch space in the asides directory of the workshop 6 subdirectory the whole human primary assembly, but all the chromosomes together, the whole 3 gig or whatever nucleotides. It's about a gig file because it compresses fairly well. ACGT is only 4 letters, and so you only need sort of 2 bits instead of 8 bits to say one of those characters. The soft masking uppercase lowercase gets a little more than that, but anyways, so it compresses decently. That's why it's smaller than the number of base pairs. But here's a little command to here. Just copy this so we don't have to type the path to this thing. We'll have a shell variable for the path to this thing here. If you want to see what that file looks like in situ, there it is. And there's the number of bytes it is. So it's 941 meg in the decimal measurement system. We could of course look at it, but we're not going to sit here and page for this whole thing. It's huge, right? So one way to do that is to unzip it on the fly, but leave the original alone, and just ask to, using grep or egrep, the caret here means at the beginning of line, and the greater than sign means look for the greater than sign. Since there should be no greater than signs in the meat lines of ACGT ends and so on, you don't really need to restrict it to the beginning of the line. But anyway, logically, you're asking for a greater than sign at the beginning of the line. That's really the definition of what the headers are. And so this will scan the file and throw away all the data lines so that we can just see the headers, so that at least we can see what's the subset of sequences that's contained in this file, which is very good. And does it have the unplaced contigs or not? Does it got the mitochondrion in there? Who knows? Well, at least we'll kind of be able to scan this and sort of see. And it takes it a while because we're going through 3 billion lines, 3 billion characters of text here is flowing through that pipeline. There we go. Okay. So all the big guys came first. You can actually see the order that they're in the file. It looks like somebody sorted it by name. But one of these great problems with sorting by a string, if you consider these as numbers, this isn't the right order. But as a dictionary, normally in strings, we sort by the dictionary order. Start at the first characters. If those are different, take the one that occurs earlier. If they're the same, move to the second position. And space, space sorts before all digits in the ASCII order. So that's why 10 sorts before 2 space. And so that's sort of not so great in terms of numerical order, but it's what makes sense for strings. So you're always battling this in computers. If you're sorting by strings and there's decimal numbers written around, they're always going to get placed in the wrong order. But anyway, very common. You will find genome files that somebody's taking care to actually order them in the numeric right way, and you'll find these that are just sort of machine sorted without really that nicety. You can see it's got the mitochondrion, MT, the X and Y sex chromosomes, and actually this one does have all the unplaced junking stuff. So there's KIs, session numbers, GLs, and so on. Anyway, that's a little aside. Okay, so now we got to index the reference here for BS-Bolt. So we've got our genome FASTA, genome FASTA here. It happens to have only a single entry in it, but BS-Bolt would be happy to work with more entries in a single file. Let's remind ourselves what the parameters are to this thing. Okay, there it is. And so now we can just sort of fill in the command just looking right at this little reference, right? Okay, so let's just go down the options one at a time. Okay, capital G, that's the path to the reference genome FASTA file. It's sitting in the current working directory, so I can just name this thing. I can either copy and paste in my GUI. It also seems to be one of the few things that starts with an H, so I can just hit H tab completion. It's ambiguous at this point, but actually the prefix there is exactly what we need. Okay, what's the next option? DB. And that is the name of the directory that is going to be created that will contain the index. And so let's name it something sensible. Okay, so this is a BS-Bolt index. And what are we indexing? We are indexing GRC H38 chromosome 21 from Ensembl release 103, the softmax version. Although the softmax shouldn't really affect the index. But anyway, that's a descriptive name of exactly what this index is for. And the rest of the parameters, as we kind of went through, are either not relevant or we can leave them as they are. So let's just run that. Okay. And it's grinding away. This will not take very long on the size of genome we have, but it takes more like 2.5 hours if you're doing the full thing. I think the index for the full human comes out to be something around 18 gigabytes on disk. Okay. We get a short break while that's working. You can see that the length that is being indexed here is actually like 4 times the length of the chromosome. That's because it's doing the forward and reverse strands, but also once you do the bisulfite conversions, there's sort of 2 more strands to index. So there's sort of the C to T converted version and the G to A converted version. So we got 4 times the original length to index. BWT is the Burroughs-Wheeler transform, SA is a suffix array. These are all things related to fast indexing of large strings of text. The computer science literature on rapid indexing and searching of text is a very well-developed field. And it finds good application in sequence alignment for high-throughput sequencing. Okay, so we're done. Okay, so we've got this new directory here that contains the index, and it's full of goodies that only BS-Vault really needs to know how to interpret. We can just leave those alone. And so there it is, we're ready. If we wanted to, document our commands, we could copy and paste this into a little text file, right? So I could grab this. This is the output. So I just copied it into my GUI clipboard. And what did I, just for consistency, maybe I didn't, I don't know, maybe I didn't, whatever, don't always name these consistently, but there it is. I just pasted it, hit Control-D to add an EOF, and now I've got a little log of how that was made. Okay, we are ready to do the alignment. Okay, so let's look at the align module and what is. Oh, it's got a lot of arguments. Okay, can we fit them all on screen? Yeah, I got to make the window bigger, but there's all the arguments. Okay, so what do we got here? Well, there's some basics that are here. So that is where you put in your input FASTQ, and a GSeq one is just fine. This is only, the F2 is used if you have paired ends, and this is the reads for the second end. We don't have paired end data, so that we just leave alone. There's the name you want to use to construct the output file name. So the main output of this is going to be a BAM file, which contains the alignments in a packed binary format that we'll look at in a second. And this is the name of the output you want the output BAM to have, except without the .bam extension. It's just sort of the base name with the prefix, as they call it. How many threads, how many CPU cores you want the aligner to use. So that makes it faster, and we'll manage that in our job submission business. We need to tell it what index we want it to use, which is the index we just made. Here's some stuff for injecting metadata into the BAM file, which we can not worry about. There's some stuff here for dealing with ambiguously mapping reads, multi-mapping reads, which we can leave alone. These are the parameters that control the scoring model. How much a match credits in scoring is, base mismatch, penalties for mismatch, penalties for indels, penalties for extension of gaps once you have an indel going, penalty for not aligning end to end on the read. So if you leave little bits of the read unaligned on the ends, penalties for that. So they're all this. There's a scoring model under there, and you can tweak it. We're going to leave it alone. This is where you tell it if the library is a modern directional library or one of the ancient undirectional libraries, where even the first end of the library contains all 4 bisulfite strands. There's some stuff related to non-conversion filtering, which we're not going to get into, and we're just going to leave alone. Okay. So this number of CPU threads is the threads that are involved in compressing the output BAM file. Usually you don't need many of those because usually the aligner stage is the bottleneck and not the output compression stage. So this, 1 or 2 here is fine. The more important number here is this one, which is the number of BWA threads. BWA is the aligner being used internally inside BS-Volt. And so this is really the number of threads that are crunching on alignments, taking, each thread takes one read at a time and is working on finding out where it goes. And you can have multiple threads working in parallel. This is related to how the initial tentative matches are found, the seed alignments. There's some heuristics. These are basically now all heuristics for how the alignment is going to make itself faster by making some compromises. And these we'll just leave alone. Okay, these are all tuning parameters, just like BLAST has tuning parameters for similar kind of stuff. Yeah, we can do this. Our reference has no alternate contigs. That's not relevant. This is the minimum score for an alignment to make it to output. The alignment model is probably balanced in such a way that 0 ends up being sort of at the point where random chance, you're breaking between negatives, which is random hits, and positives, which are statistically significant hits. So this is currently set to 10. I mean, it depends on the scoring model, what's parameters, what's reasonable there. And then this is a parameter that's relevant for paradigm libraries, although these days usually just let it internally, it'll align a few reads to estimate the mean and standard deviation of the fragment length, the distribution, and then it will use that. It'll kind of auto-calibrate. And it's not relevant anyway for us since we don't have paradigm data. Okay, so that's a walkthrough of all the options there, and there's not many of those that we really need to set then. So there's really only sort of like these 4 that are big for us to set. Okay, the rest we can kind of accept as defaults. Okay. All right, so now we got to work our way towards building a command line that's going to achieve alignment of a file for us, and then this time we want to use the job submission system, because typically when you're doing alignments, you got big files, many samples, and you got to use Hoffman. You can't do it all in these interactive sessions. So let's build our way up to a nice set of batch system using scripts and so on. But we got to work our way through it. So basically, after consulting the documentation for the built-in documentation for BS-Volt, we know that the command we want to run, the template looks something like this, right? So we're going to BS-Volt align db whatever the index is. F1, we give it the fastqgz reads and some output prefix, which is going to be based on this file name. And then we also got to tell you, we're going to be using multiple cores in our jobs for speed. And we're going to have to tell BS-Volt how many cores it should use. So filling that in with an actual example, right? So, so this is one of the fastqgz files that we're going to apply it to. It's just sort of just to see what it would look like expanded out. This is the name of the index we just made. That's the name of the reads. And the directory will be, the script will be running in is the same directory, the day 2 directory. So relative to this point, the reads are in the stage 3 place, right? So the paths to that stuff is in stage 3. This isn't really a stage 4 to 5 so much as it is a stage 3 to 5. But, there's a subtlety of whether you sort of want your stage names to really reflect the actual dependencies as to what subset of stages provide the data to number the next stage, or you just number them n-1 to n, even if 1 to n-1 isn't all the stages that are relevant for making stage n. Anyway. This is the output prefix. We'll basically make it exactly the same as the input. We'll lop off the fastq.gz, and then we'll add some description about what this one's doing, which is we're BS-Volting to this reference. So we'll embed that into the file names as sort of some breadcrumbs. And we're going to probably use 4 or maybe 3 cores. I think I started with 4 yesterday, and after seeing how long I was having jobs starting with 4, I decreased it to 3, I think. But maybe at this point I was still 4. And then we're going to log the output of BS-Volt into some files. We're going to capture the standard output. That's what UNIX calls screen number 1. And we're going to capture the standard error, which is what UNIX calls screen number 2. Okay, so that is all good. We are going to isolate the BS-Volt standard output and standard error into its own files because, 1, that will connect the standard output and standard error, which is important if there are problems. It'll let us easily know which original source file had the problem because we've embedded the original source file into the name of the log file. That's important. Because the standard output and standard error log files created by the job submission system will essentially be assigned random numbers which are related to the job ID on the system, and it's going to be difficult to connect those back to the original files. It's possible to do, but it takes some work, and it's just easier to have, if there are errors at this stage, we'd like to know more directly which file they're connected to, and also there is some statistical information that BSbolt will print to, I think it's standard output. And capturing that for posterity and so that we can examine it later, we want to be able to do that easily and connect that to which file has which stats and so on. So this is all going to enable that. And then if there's stuff at the job submission level, kind of serious problems at the job submission level, we'll be able to easily see those in the job submission standard output and standard error. But we kind of isolated out all this stuff we're expecting to come out and attach that more directly to the input file. So that'll become more clear when we actually run this stuff. So using some shell variables, we can make this generic to all of our samples. We can, in GZFQ can be a shell variable that contains the name of the FASTQ input. We can construct outbase, which is this thing, the output prefix, by just lopping off the .fastq.gz, like we said, and tacking on the stuff we want using the shell built-in substitution there. And then our BSbolt align command looks something like this, right? So we can basically fill out most of the parameters now with our shell variables, okay? Also maybe we'll want to make we could leave the T-4 and hard code it into the script, but maybe it's better to, what's -T for? You got to, you'll come back to this in 6 months and you'll be looking through the commands and you're like, what does T4 do in BSbolt again? Why don't you just name it in a shell variable or something that makes sense, right? We'll just call it alignment CPU cores and set it to 4, and then in the command we can do -T, whatever. And now it's immediately obvious going back to the script at any point in the future, long after we've kind of written this, what this purpose of this was. Okay. So this is we've got this point built up. The next issue is that once we have our alignment BAM file, it's going to have some issues that make it not ideal to use with downstream programs such as IGV visualization. So one thing that's going to happen is that the output order of reads, reads are producing alignments. So the alignments, i.e. reads, as they come out in the output BAM file, are essentially in a random order. It's mostly related to the order that they are in the input file. Now, because they're not likely to be exactly the order that they're in the input file, because we're going to have BSbolt work on things in parallel. And so it's probably going to read little chunks of the file and then give chunks to each of its little thread workers. And each thread worker will be generating results in parallel, and probably the output file gets the outputs in interleaved order, possibly at least. I mean, this is common. I don't know if BSbolt takes the pain to sort of unpermute things as it produces its output. Some of the aligners will do that. Sometimes it takes a command line option fast to do that because it takes more work than just, if your order doesn't matter, then why do the work? So it's likely that the output BAM file order is going to be somewhat related to the input read order, but the input read order is completely random, right? It's related to the physical order that the nanowells were on, or it wasn't nanowells in that day. It was the but anyway, on Illumina platforms, whether it was a pre-ordered flow cell or whether it's a modern ordered flow cell, the read orders are related to the physical positions on the flow cell, the physical substrate on which the sequencing chemical reactions occurred. The order of the reads is related to their physical positions on it. It's ordered in a particular way relative to their physical positions. So in terms of the reference genome, those fragments are just completely random orders, right? We don't expect any ordering that makes any sense to be in read order. So most programs that want to read BAMs often have a requirement that the alignments be reordered in the BAM to, first of all, group all the reads that belong to each chromosome together. And furthermore, within each chromosome, that the reads are essentially in order as you scan the chromosome left to right. So usually most things are quoted relative to the plus strand, and usually the plus strand is numbered 1 to n, the base is the plus strand, 5' to 3'. Sometimes it's 0 to n-1. That's always an issue in bioinformatics, what the numbering system is. Do you start at 0? Do you start at 1? And when you say A to B, is the A included or excluded? Is the B included or excluded? Those are the most common flavors. And then sometimes when people quote numbers on the minus strand, they count the minus strand from the 5' end as a 1, and some people count the minus strand from the 3' end as a 1. It depends on whether you want to quote everything against the plus strand or whether you want to start to quote everything against 5' ends. Anyway, so there's all these combinations of coordinate numbering in bioinformatics, and each file format will have its own conventions, whatever. There's a few that are common, and of course the BAM spec is all completely defined. It's not ambiguous, but it is what it is. Anyway, so in the BAM file format, there's one essentially column that declares the chromosome that the alignment is 2, and another column that declares the leftmost position, the smallest numeric coordinate of all the aligned bases using the plus strand as the reference for the numbering system. So it's the leftmost aligned base on the plus strand. So a lot of programs require that the reads be ordered such that the chromosomes, all the reads for chromosomes are grouped together. The chromosomes are in string sorted order. The names of the chromosomes are in string sorted order, and the positions within a chromosome are in ascending numerical value for the leftmost aligned base that's quoted in the plus. So that's called a sorted BAM. And SAMtools is a program that can take us any BAM file and resort it into the correct order. So we're going to need to do that because, for example, IGV has that requirement. And furthermore, BAM, which is a compressed file format that's actually based on gzip, normally gzip files can't be randomly accessed. To read in the middle of a gzip file and find out what the uncompressed data is at a random point inside the middle of a gzip file, essentially you have to start at the beginning and decompress the whole file to that point. And for BAM files can be 100 gigabytes long. You can't do that. That takes forever, and you just can't, not workable. So you need some way of randomly accessing uncompressed data in this file. BAM is based on gzip, and in a way, gzip will uncompress the BAM file, even stock gzip. But the gzip allows extra data to be embedded in certain program-specific fields. And using those, BAM can sort of inject extra pre-computed data into the gzip file, which then another file format called BAM alignment index, can extract information from that and put it in a little sidecar file that lives alongside the BAM file. And from that embedded data inside the BAM and the index file, it enables programs to randomly access uncompressed data in the BAM, which is great. So you get the benefits of compression, which is crazy good for BAM, because SAM files, which are essentially uncompressed BAM files, are plain text, they're usually gigantic compared to BAM, like 10x or more larger. And so it's really something you really don't want SAM files around because they just take up so much space. And BAM compresses, the BAM compression is quite effective and very useful. You don't want to really give up an order of magnitude on your disk space and IO time, network transfer, and so on. So we want to do this, right? So we want to sort the BAM, and then we want to index it. And indexing will produce a .bam.by file that just lives alongside the BAM file. And those together should be considered a pair and always move together. And IGV will want that by file as well. Because in IGV, you're going to be browsing around in random places, and it needs to retrieve all the reads that are in the window that you're viewing. And the way it does that is with the by file. And the by files are very tiny. So that's the story with that. We need to use SAMtools, which is another little package, and which is actually already available on Hoffman. So if you look at module avail, and maybe, I told you it's called SAMtools, and maybe just search for tool. There's bed tools, QTL tools. There's SAMtools, right? So there's a couple versions, some old ones. The current one is 1.9. That's the default. So you could just say module load SAMtools without a version. I generally like to specify the version because you generally want to know what version you use later to do an analysis in case there's some aspect of it that changed in the meantime, and you need to know whether the version number matters. And so you sort of need to know how to record which version number you're using. So you might as well always specify the version. Okay, so we're gonna activate SAMtools, and with that we have a program that we can do all kinds of transformations on BAM files and SAM files. And it turns out there's also CRAM files, which are even more compressed than BAM because they use the knowledge of the reference genome to sort of compress out all of the data you can with that. Anyway, there's all these little modules of this program too, and one of them is the SAMtools sort, and that'll sort a BAM file, make a new BAM file, and there's also SAMtools index, which will take a BAM file and produce a by file for it, and that's what we're going to use to do those two jobs, which are sort of like housekeeping jobs that we have to do after producing a BAM file. Okay, so yeah, so we just did a bunch of slides, and looking at the command line help for these SAMtools sort and SAMtools index, the commands we're going to need are something like this. SAMtools sort, it also can do multi-threaded to be faster, so we'll give it 4 or 3 threads or whatever the heck our job submission environment is going to launch our jobs with. And there's how much memory each thread can use. So 4 and 5 gigabytes on memory and 4 threads would use 20 gig of RAM at most. Which is great for sorting, because the way sorting works is you read a whole bunch of stuff into memory, which is fast to random access. You can sort those in memory. And then if the whole BAM file doesn't fit memory, you push that out to disk as a temporary file, you grab the next chunk and so on. And so you turn the file into a bunch of sorted chunks on disk, and then there's a merge process. And merging sorted files is quite fast. You can do that with just a linear scan of all the files. And so that's how sort works. And so the 5G is sort of the chunk size that each of the thread workers works on. There's the input BAM file, and that'll show you name the output BAM file. And Ensembl index, once we produce the output BAM file, you just say Ensembl index, and then for some, who knows reason, it's an @ sign is the name of the command line parameter, which is such a scary shell character. You might as well single quote this so you guarantee that, you'd have to look up the rules for shell, whether that's really going to work without the quotes or not. You might as well just quote it. But anyway, yeah, and I've tried it. Ensembl index does not take double dash threads like the rest of Ensembl's stuff does, but whatever. It's just history or whatnot, some oversight. So anyway, this is the way you tell threads. It's the same thing. I think actually the Ensembl sort takes the @ sign version as well. It takes the longer double dash versions of options. And in general, I prefer the longer double dash versions of options because they're more self-documenting when you read your command lines later. But they're just longer to type, but typing's not really that much of an issue. So anyway, so this is basically threads 4, right? So index can also use multiple threads, and that will produce, if in.bam is given to it, it'll produce an in.bam.by, which programs like IGV and so on will need. Okay, so this is now our plan. Our plan's got slightly elaborated, right? We're going to run BS-Seq to create the alignments and make the BAM file of alignments. Then we're going to sort that to make a new sorted BAM file, and then we're going to index the sorted BAM file to get a sorted BAM by file. All right, that's the plan. And now we need to wrap this up for the job submission system on Hoffman. And we're going to do 2 scripts for that. There's one script, which is the work script, which is going to do this actual work. And that will be run once for each input FASTQ file, right? And then there's the submit script, which will handle the business of configuring the job system on Hoffman to do all those work scripts, right? It's going to do stuff related to job control, communicate the resource requirements to the scheduler, and it's going to communicate the list of FASTQ-GZ file names that we want to process into the work script. Okay, so, all right, so that's the plan. We need to write these 2 scripts now. So let me now give you those 2 scripts. Okay, so I'm gonna just ignore that. I'm gonna switch to my place where no. So give everybody read and execute permission to the stage 425 scripts. Okay, so I just fixed those. And so you need to copy them now to your place. You're still looking at your day 2 directory in your personal space. So now you've got to go and pull those into your space. So that would be like this, and use some tab completion, day 2 start state, and stage 425 star.batch. Save and put them in the current working directory. Okay. I will actually put the VI here because for me, that's going to be an overwrite because I've already got them here. And you'll see what CP does when there's an overwrite condition, what that little -I, that little I gives you. And it says you're about to overwrite in the current directory this file. Do you want to do so? A Y and enter would do it, and an N enter will skip it. So I will say N and N. And so my copies I had here already will not be touched. If I had said Y and Y, they would have been replaced. You will not get that message because you don't have those files there. Okay, so is everybody able to grab those now? Okay, great. I'll give you a minute to get them. So this is what the 2 scripts look like. This is a work script. This is job submit script. And they probably look pretty scary right now, but they're not that bad. There's a lot of functionality in here, which is quite useful. So the way it's going to work is that the launcher script, the submitter script, we're going to give it as command line arguments all the FASTQ files, all the FASTQ-GZ files we want it to process. Okay, so that's what's going to come into that thing. Okay, so here I'm just setting up some shell variables for kind of the parameters of the analysis to kind of give an easy and documented place for where the parameters of the analysis are. So the job submission system on Hoffman, you can give a name to jobs, which shows up like in the queue status, and it shows up in the log file names and so on. So it's useful to give something not too long, but descriptive there. So this is the alignments of BS-Bolt. That'll be the job name. And I decided to use 3 cores per worker and 1 hour per worker at most, right? That's a limit. Doesn't force it to take an hour. It just says can use up to 1 hour. After 1 hour, it would be killed immediately. Each core is going to get 6. We're going to ask for 6 gig of RAM allowance from the scheduler. So that's going to be with 6 times 3. That's 18 gig of RAM total per worker. The sort, I feel, is a little dangerous, the SAMTools sort, to ask for SAMTools sort to really push the limit, because you don't really know how the rounding works and so on. So let's lower that to 5 per core for the SAMTools sort, just to give us a little margin. So going over is bad, right? Going over is death, right? With no progress. So let's aim a little under. And then let's also parameterize the, this is the BS-Bolt index that we're using, the reference gene, essentially. So, okay, so now we've got some shell variables to abstract away those parameters. First thing to do is that it turns out that the Q subcommand for job arrays will not construct a job array with 0 entries in it, right? It's like, why are you bothering the Q system if you have nothing to do? So I will check in the script here whether I have no command line arguments. And if so, I'll give a little usage message and then die. That just allows you to run this command with no options. We've often been doing that to get help out of commands and stuff like that or whatever. It's going to happen sooner or later. So let's just deal with that case. And it gives us a little place to sort of give a reminder of what the parameters of this thing are supposed to be anyway. All right, so here's really the only command of any, that does really anything in this whole business. And it's this Q sub command, which I split on 3 lines with 2 line continuations. Okay, so let's walk through the little bits of that Q sub command. So here are some parameters that are just sort of general that are useful for every job. The CWD, and you can see here that actually Q sub is a more modern command and doesn't follow the Unix convention of long options taking double dash as a start. So that CWD is actually not a -C -W-D. Should really be a double -CWD, but that's just not the kind of society that Q sub comes from. So this is a single option, and it means that the working scripts will have their current working directory, the thing you get when you PWD. They will get the same one that the launcher gets. All right. So when we call the launcher script, whatever our current working directory is, that's inherited by Q sub. And then whatever Q sub gets will be pushed with the CWD to the actual worker scripts as they start. So everybody will be in the same working directory, which is great. That's generally what you want. So that means we can use relative paths everywhere and it will all just work out. The -R no means that the job should not be auto requeued if the job system detects errors. We just want it to fail and die. We'll deal with that in case it happens. We don't want magical auto rerun attempts. The J no means that at the job level, manager level, we want the standard error and standard output logs to be 2 separate logs. The MAS sends you an email if the job gets aborted, rescheduled, or suspended. The A is the aborted, or maybe the aborted, rescheduled. The S is the suspended part. There's other letters you can add for. You can have an email you when jobs start, when jobs stop, or whatever. Usually the start and stops get pretty annoying pretty fast. And so usually I only do the emails for the ones that kind of go weird. So the A and S, like somebody hits a time limit or whatever, they're killed, that'll be an abort and you'll get an email with the MAS. I have no idea what Hoffman's default is for your email address. If it sort of knows when your account was created, if somebody kind of put that information into it somewhere, it's easiest to override whatever it is. Anyway, you all have probably 19 email addresses anyway, and you maybe want to control which one it goes to. And for that, you can create a single line text file in your home directory called .forward, and just put your email address in there as a one-liner. So for me, the contents of that file are my email address, and that should override any account information that's put into Hoffman by default for you. Otherwise, this may not work. But anyway, that's that. And then the -N is the job name, which we're going to be aligned BS-Bolt. Okay, so that's that part. Next part is the resource requirements, which are, we're going to ask for so many cores, three cores per worker, 3 CPU cores, and the runtime is going to be 1 hour, that's this, and the RAM virtual memory is going to be 6 gigabytes per core, so 18 gig total. That's what the worker's allowance is. Each worker has that allowance. Okay, then the rest of this here at the end, this last line. Yeah, all right, so yeah, so we've already kind of talked about this at the beginning. You know, the higher you make the resource requirements, you know, the faster you could sort of, you can make it work, but you're going to wait longer because the subset of the cluster that can satisfy those requirements at any moment is going to be smaller. And eventually the requirements get so high that you know, either the wait is very, very long, or essentially the wait becomes forever if you ask for requirements that actually don't exist on the cluster. So that's the fight for why you can't make them too high. Of course, if you make them too low, then either your jobs finish very slow or they get uselessly killed by hitting the memory or time limits. And so there's a fight between those two things. And experimentation is often required to sort of figure out what a good balance is. Okay, so this last line here is basically the command line we want each worker to run. And that is the name of our, that is essentially just like we would kind of do it on a command line. We don't really need to give the path because it's all local and this isn't really a literal command line, but QSub will kind of do the business for us. We don't have to do the little / on the front, although it wouldn't hurt either. The name of our worker script. So this is the command that QSub will run. And then we can give the command line parameters to that script. So we're going to give, to make this script a little more configurable too, we're going to give it the number of cores because it needs to feed that into various parameters of BS-Bulk and SAM tools. And we're going to give it the number of the RAM that each sort thread should use. That's the second parameter, and that goes into the SAM tool sort, one of the options. And we're going to give it the BS-Bulk index so that we don't have to like embed that into this. We can just have this one place where sort of all the main parameters for the analysis are sort of over here. We don't distribute them all over the place. And so we'll pass in the BS-Bulk index as well. And that leaves all the FASTQ-GZ file names to give to this guy. And that's all the that's all the command line parameters to this script. That's all the FASTQ-GZ we need to do. So we need to pass now, you know, this is the first argument, second argument, third argument. From the fourth argument onward for the worker, those need to be the FASTQ names given to this guy, you know. And this little $@ is a little shell magic with double quotes there that expands into exactly the same list of command line arguments that I got. So it's a way of sort of, you know, just passing through what we got onward. And this, you know, if some of these command line arguments had spaces in them, it would preserve them in someone. The number of arguments this creates is exactly the same number we have with all the white space and everything preserved exactly the right way. So that's how that's done. And the last business is the magic for how we get our worker script to know which FASTQ file of that list it's supposed to work on. And the way it works is that we're using what's called a job array job in the UNIVA grid engine, Sun grid engine speak. And so that's what this -t 1 to something :1 means. This $sign is the number of command line arguments we got. So if we gave it 3 FASTQ files, then this thing $@ expands into those 3 FASTQ file names. And this $sign becomes a 3. And so essentially we're saying the job array is jobs 1 to 3 stepping by one. And so essentially we want there to be 3 workers. There's going to be worker number 1, worker number 2, and worker number 3. And that is how the worker is going to know which of the FASTQ files it's going to get to run. It's going to get its command line arguments, all of them, but it's also going to get a number that tells it which of those is the one that that particular worker instance is going to be supposed to do. And the way that's communicated is with something called an environment variable. And environment variables just turn into shell variables of the same name inside a shell script. And the environment variable that's magic is called SGE_task_ID. Hoffman uses the UNIVA grid engine, but it was formerly called the Sun grid engine before the fork. And so that's why that the SGE is here. So that's the prefix of all the SGE, now UGE related environment variables. And so this task ID becomes the 1, 2, 3 in turn from here. And that is how this script knows which of the input files it's supposed to work on. Okay, so now we can look at the worker script. Okay, so it's a bash script too. It's going to take the first command line argument is what we gave here, numcore. Second argument, sort for core degree, that's here. Third argument, pass the BS-Bolt index, that's here. Then the one or more remaining arguments are the path names that end in FASTQGZ. That's what we're supposed to work on. Then there's this environment variable, SGE task ID, which gives us the index into which of those fourth onward arguments is the one we need to pay attention to. Okay, so we're going to now extract out our command line arguments to get ready to figure out which is the FASTQ file name we need to figure out. So I'm going to pull out the first argument to us, this with the worker, put that in numcores, that takes care of that one. And then the shift is a little shell built in that's kind of cool. What it does is it takes all the command line arguments to the current script, throws away the first one, and numbers all the remaining ones down one. So the first argument's gone, the second argument is now the first, the third is now the second, and so on. And so basically we kind of captured the numcore's first argument in a named shell variable, and now it's not in the command line anymore. And now it's as if it was never there in the command line argument number. So now the new first argument would be the second one originally, which is the sort for core GB. So we extract that out, shift that off. And then there's the DB, which is the path BS-Bolt. That's the third one. We pull that out and shift it off. Now we shifted off the first three command line arguments to the script, which is exactly those. So now the first argument that's left is the first FASTQGZ that needs to be done, which is exactly the same numbering that this guy has. And it's exactly the numbering that the job array jobs are numbered in. So now everything's consistent. So all we need to do now is to look up the SJE task ID number and take the command line argument in that position. And so if I just did $SGE task ID, that would just make this be the 3. That's not quite right. I want the SGE task ID to turn into, suppose this is the second worker of 3, right? So then I want the SGE task ID to turn into the 2, but I don't want ingzfq to be 2. I want it to be the second command line argument at this point. And that is the magic of the exclamation point, which means that the variable that follows, the shell variable that follows, is not to be taken as the thing you want the value of. You take that thing, get its value, and use its value as the name of another shell variable to pull out. And that's what this guy gets. So the SGE task ID will turn into 2. This is essentially $2, which is the second command line argument. And due to the fact we've shifted off this business, that's now the second FASTQ GZ file name that we need to process. That's exactly what we want. And so then we can construct our outbase by dropping the FASTQ GZ, tacking on that. And at this point, you can sort of peek ahead and look to this. And now we're sort of set up in a situation where we can execute our plan, right? We had from a couple slides ago, wherever it was, right here, this was our plan. Basically, that plan is exactly reflected at this point in the script. Right? So that's that. How is all my stuff lining up? Is that a tab? No, actually, I'm just using spaces. Tabs are a bad idea for a lot of reasons. Python is a great example of that. If you make white space significant, there are different kinds of white space, and tabs vary what your editor says for how many spaces a tab limit to where your tab stops are. Anyway, so yeah, the best approach is to never use tabs, just use spaces. And I tend to carefully insert my spaces to try to line things up. It's just much easier to read. It kind of shows the structure of things and so on. And yeah, in reality, I really do write my shell scripts with the indentation and spacing sort of like this to line up relevant stuff. Okay, so that was a question from the chat of how the spacing and the shell script is sort of maintained this way. I've also done some syntax coloring here of the shell script to help you parse the parts here. So, okay, this echo here and this echo down here is just progress information like we've done before. There's more progress information here. We'll include in logging the host name, which is the node number, the Hoffman node name that's actually executing the script, and the name of the script the job submission system gave it. And we're going to log all of our command line parameters basically here, except instead of the full list of FQGZs, the particular one that we're going to be working on. Anyway, so we got to start, and the script reached the end guy. Then there's this stuff in the middle, which we've seen before in day 1. Which is getting our environment set up, right? Remember, we got our shell script, just like our shell scripts do, the job submission, scripts run by the job submission system come into a blank, slate. So they don't have the same environment interactive place does. So we gotta load that up. We need Python because we need BS-Bolt, and we need SAM tools because we're running that. We gotta activate those modules. And then we got to set our path so the BS bolt is there. Anyway, so this just gets our environment into the script as it was in our interactive world. Then there's some stuff related to error handling in shell scripts, which is really an advanced topic and can get very complicated. And I'm just doing what's kind of easy to do here. I sort of want, if any of these commands fail and return a non-zero exit status, I want to capture that. And that's what this set-o pipe fail pipe status equals $$ does. If any sort of these subcomponents have an issue, that's communicated by having a non-zero exit status. This will give that, and then we eventually return that same status from ourselves, but don't worry about that too much. And these double and signs, ampersands, are kind of like semicolons that you can run multiple shell commands on the same line, pack them onto the same line. It's like a semicolon, but it really runs the first command, and only if that succeeded does it continue on with the rest, and so on. It's sort of an early abort if things go wrong. So if the BS bolt sort of has, kind of explodes, then the samples won't run here. It'll just stop. Or if the first sort dies, then the index won't run. Okay, and then the final little bit here is that there's a parentheses here and a parentheses here, because I want my capture of the standard output and standard error logs from this running this little shell pipeline. I want the BS bolt and both SAM tools to all go into the same files. And so that parentheses groups this into a there's actually a sub shell that will get run that kind of handles this, but the entire output of this whole sub shell gets redirected into these two files. And so that's how we capture all three of these things all together. If we had just, without the parentheses, the redirections would just be applied to the last command in the pipeline, and we'd only be capturing the SAM tools index stuff, which is not what we want. So there we are. Okay, so that was a long, you can take a breath. That's about as bad as it gets, okay, in this workshop, I think. There's a lot of shell stuff going in there and so on, but it really gives you a nice, once you got this kind of boilerplate stuff worked out and so on, it's a nice way to work on Hoffman. So we're actually going to run now, and then we can take a break because it's going to take it's going to run for a bit on your guys. So let's get to actually running it, right? So here I am in your personal scratch space. We have the scripts ready to go. Let's first try running it with no parameters, right? We actually have a little, no, not the worker script, not the launcher script. Okay, there we go. All right, so there's our little error handling, and it says, oh, you probably don't know what you're doing. Here's a usage reminder. Okay, so, all right. The only parameters this thing needs is the list of FastQGZs to work on, which are currently the cut-adapt ones. That's stage 3 cut-adapt stuff. What does that look like? I just hit tab enough times until completion reminded me what these look like. All right, so yeah, so these are the FastQGZs here. So do all the FastQGZs in the stage 3 subdirectory, which is 6 of them. That's exactly what we want. And there we go. All right, so doing that. And that's the message from QSub, your job array, which is job number 6655234. Yours will be a different one because they have to be unique. It is an array job with subjobs 1 to 6. And it's called a line BS bolt. Now, qstat is a command that you can see what the Q is doing. The -U restricts the qstat listening to just yourself. You get any user you want, which for me is that. And it's a long entry, but oh my gosh, they're already running. That's great. Right, so that was fast. So this is my interactive session. This is the session I'm typing into right now, QR login on node 7361. Here's my node 7361. And I've got 4 cores allocated to this interactive session. The R means it's running. Here are my aligned BS bolts. Now you can see here, I got 4 of them running already. Q wait. I've got numbers 5 and 6 are waiting. All right, they haven't started yet. These guys are running. These are the node numbers they're on, and they've all been allocated 3 cores each. And this is job number 1, 2, 3, and 4. All right, that becomes that SGE task ID in the worker script. All right, so now there's this command my jobs, which basically just does the same thing as qstat-u yourself. I still got just the 4 running. Okay, qstat with no parameters will give you the entire queue. Well, that went by fast. How many lines was that? 4,132. Let's pipe it to less, and we can take a look at it. There's the queue on Hoffman right now. Look at all those R's running. That's all the junk that's running on Hoffman right now. Oh, there's somebody still in a DR. That sounds bad. So that's some interesting state. These are all running hundreds and hundreds. How many is this? It's a lot, right? These are all running. Okay. All right. We're at line 3,000. There's some 3,000 jobs that are running. And if you look at the last column, which is how many slots, you know, some of these jobs, 96 cores allocated, 48, 32. Some of these are going to be not just on one node, but an MPI multi-node jobs. You can't really get 96 cores on a single node like we've been doing. You know, here's 500 cores, you know, blah, blah, blah. Oh, here's other people with job arrays, right? And what job number in their job array they're working on. Some of their job array, here's a guy with job array subjob number 503, right? You know, so, you know, there's a lot of stuff going on. Eventually at the bottom of the queue here, queue wait. So here's the boundary. These H's are held jobs. Those are administrative hold for who knows what reason, maybe use or who knows what. QW's queue wait. So these are the jobs pending, right? Anyway, so you know, mine should be in here somewhere, right? So if I look, you know, I just search for Cocos here. Slash is how you can search for a pattern in less. And then once you have slash, you can just slash enter to find the same thing as the last one. So anyway, so back to the top, there is one of my runners. There are some more runners, and down here, these are my two waiters. You can kind of see the here's some friends here that are running the same thing. So anyway, so that's you know, that's so you can poke around. So it's been a little while longer. Still only got 4 running. Anyway, so this is going to take a little bit. This is doing serious work at this point. So we can, you know, let that go. We can kind of poke around and see what's happening. At least I got some started, so you can kind of see how this is going to go. These are the log files being created by the job submission system. Okay, so this is my job name here. The E's are for standard error logs. The O's are for standard output logs. And there's also PEs and POs, which are from the parallel environments since we're doing these multi-core businesses. That's the PE share stuff. The suffix here, this is the job ID, 665234, 665234. And this is the subjob number, the SV task ID. So I got 1, 2, 3, 4 are launched, 5, 6 haven't launched yet, so no logs from 5 and 6 yet. These are the logs, and you can see some of these are empty. The P is empty. Empty is good, right? Because empty means no errors, so that's good. Now, there's a little bit in these, and there's some of these O's guys. Let's take a peek into those guys. So let's look at some of these. All right, so let's see what these error guys are doing. So I can just cat all of them, right? Whatever. Well, no surprise there. That's just some kind of useless information out of the module stuff. So that's completely normal, nothing to worry about, no problems there. Not worried at all about that. What's in the standard output logs? All right. Well, that's our logging from our shell script. We got our worker script that has that doing and reach the end kind of stuff. And so these are the logs. I'm working on this FastQGZ file against this BS-Seq database, and I'm making this output. And I'm running with 3 cores, and I've got 5 gig of RAM that I'm going to use when I get to the sort. And the shell script got put in this place by the job submission system. And I'm running on node 6361, and I got to this point at this date. Right? So that's all great info. So there we go. So and obviously none of them have hit the bottom yet. They don't have the finish message yet. So they're fine. Nobody doesn't look like any errors yet. Oh, this file just got bigger. Maybe this one's done. So number 1, maybe number 1's done. So, oh, and indeed he is. So he reached the end, and there's nothing in between, which is great. That's exactly what you want to see. We have no kind of job level errors, right? The analysis is one thing, right? Remember, we redirected the output of Bolt and SAMtools into other files that are parallel with the input FastQs. But at the job control level, there's no serious problems, all right? All the commands were found. We started BS-Bold, okay, and so on. The Sheldon said permission denied, or can't find it, or whatever. Anyway, this is great. So that's good. Let's take a peek into the stage 3 directory, which is where the outputs are going to be accumulated, same places where the inputs are. All right, what's going on in there? All right, so these FastQGZs, those were the input files. Well, it looks like number 5 must have launched here because I've got some output files for number 5. Number 6 looks like hasn't launched yet. The order that the job order should be the same order as LS does, because the way the shell expansion does here, this expansion is essentially the same order that LS will list them in. So we kind of know this is number 1, 2, 3, 4, 5, 6. It's another advantage of doing it this way. What do we got? Okay, these are our log files, our standard error and standard output log files. We'll take a look at those. Here is the BAM file out of BS-Bolt. Here is the sorted BAM file out of the SAM sort, and here is the sorted BAM by file out of the SAM index. So that's all looking good. Why don't we look at some of these standard error things? All right, what's in here? Let's just do all the standard errors right now. So, that's just some progress information from the SAM tools sort. Nothing to worry about whatsoever. Okay, what about, I said number one was done, right? So we can look at this standard output for number one. This is the analysis level standard output, right? This is the stuff we captured from standard output of BS-Bolt and SAM tools index and SAM tools sort and SAM tools index. And so this first business is from BS-Bolt. In fact, it's all from BS-Bolt here. Actually, what do we got? Well, there was some progress information. This is, I ran BWA with these parameters, and then BWA gives this progress information. And actually, in the end, there's some useful stuff. Well, the alignment took 4 minutes, but here's some basic statistics out of BS-Bolt, which are kind of useful. 98.7% of reads mapped, 56,000 of them mapped bisulfite ambiguously, not BS uniquely. There are 47,000 unmapped reads. This is how many reads it worked on. All the reads aligned to the C to T conversion, which is exactly what we expect from the first end of a modern directional library. Nothing mapped to sort of the two PCR'd up post-bisulfite conversion strands that an undirectional would, or the second end of a paired-in directional library would do. This is all looking good. The mappability here is a little artificially high. It's a lot artificially high because I constructed these inputs by doing a full analysis run to the whole genome, picking out all the guys that align to chromosome 21, and then giving you essentially those reads. So in a way, all of them align. I'm not quite sure why some of them don't here. It's something we'd have to look into it. Bisulfite ambiguous means that relative to the C to T confusion, if you ignore all possible methylation states that the read could have had, they're not all, the sort of the unambiguous, relative to the ambiguity, relative to the variance of the read that could have happened due to bisulfite conversion, there are multiple places in the genome that this read could go. So yes, so it's the correct sense of uniqueness, of uniquely aligning, adapted for the bisulfite assay. Anyway, yeah, so essentially they should all align. In real experiments, you're going to see all kinds of numbers there, and it's sort of hard to say. I think at some point we get to it here. I talk about it down here. Essentially, it's best if you can find an expert that's already doing bisulfite WGS or RBS or whatever you're doing with a similar prep protocol to you, and it's in the same organism as you with similar biomaterials to you to find out what's normal. If there's a significant amount unmapped, you should really try to find out what it is. And that may mean taking small random samples, 10 reads, 20 reads, whatever, of that underlying stuff and going to NCBI and our web blast and trying to figure out what it is. Maybe it's some kind of unexpected contaminant or something. You got to be pretty loose with the alignment parameters and blast in there because you got the bisulfite kind of mismatches, which aren't helping. They're going to induce mismatches to a non-bisulfite aware aligner like blast in. But anyway, yeah, if you have high on a line, you should really try to figure out what that stuff is. For example, you need to change you need to augment your reference, anyway, that's sort of just depends on the details of your situation. Okay, so, maybe, let me check, I haven't checked my jobs in a while. Oh, okay, we got numbers 1 to 4 have finished, they have disappeared out of the queue, 5 and 6 are running. So we are making progress here. All right, let's keep looking at, okay, syncing with the slides here. We looked at the log files from the job control system. We looked at the log files from our shell scripts themselves, and we are progressing to looking at the actual output files, right? So each original FASTQGZ turned into this collection of 5 files, which we've been talking about. And we kind of looked at the standard errors and the standard outs. We just now we're looking at the standard outs, right? So that was this standard out for this one guy, the number 1. And and what's next to look at? Yeah, we talked about that. All right, yeah, so a few reads. Yeah, okay, so like there are, we got you know, if you take the align numbers, right? I mean, it's almost the same as total reads input because the mappability is so high. But you know, if you take this, which is like 1.7 million plus this, which is like 2 million, whatever, you add them up and you get a 3.7 million about, right? So that's how many reads we got aligned. Our current reference genome, Chromosome 21, is only about 40 megabase pairs. 40 megabase pairs meaning 80 meganucleotides if you count both strands separately, right? So C's only live on one strand, not both strands. You need to pile up on both strands to kind of get the average coverage. We have 64 nucleotide reads that we put in. Some read bases will be low quality and inconsistent with bisulfite conversion, and some alignments won't be end-to-end and so on. So, you know, we're not really going to get full 64 bases out of every aligned read, but 60, 62. The average will be close to 64. So we got 3.7 million mapped reads, let's just round numbers, 60 nucleotides. We're going to get coverage out of each of those. And that multiplies out to 222 meganucleotides we've got of aligned read coverage. And so if you point to a particular random genomic base on a particular strand of our reference, which we've got 80 meganucleotides of reference, that means we're only at about 2.8x, it's called 3x fold coverage, average coverage. Now, because the coverage will be highly uneven, some areas are going to be like 0 and uncovered, and other areas will be more like 5, 10 even. You know, it's going to be pretty uneven because remember there's PCR involved in this. ACGT composition is all over the place, and so the biases in location are going to be pretty high. If you are considering CPGs and are willing to combine the two symmetric sites into one site, you can double the coverage, and the average would be more like 6 there. So anyway, so you know, we're kind of, we're not too good at the individual site level. You know, we're going to have to pool sites to do much statistics, but we're also kind of right at the threshold. And for WGBS, this may not even be that unusual. You're at a coverage level that's just around here. Maybe 10x is a better level to shoot for on average for WGBS. But it's basically as high as you can afford, right? So, okay, so there's that. Now, since we are kind of comfortable, well, let me see if my job's done. Well, I still got these stragglers. How's everybody else doing? How many, send me a chat for how many jobs you've got done and how many you got running and how many you got waiting, 6 minus the other 2. I can sort of answer that question by doing QStat for everybody and grepping out ALN BS-Bolt because you've all called the job name the same thing, right? So there's everybody running something called ALN BS-Bolt right now. So, okay, so we got one guy where all of your jobs are waiting, one where you got 2 waiting and 3 running and 1 done. And I've got 2 running and 4 done. All right, if you got issues, try to look at the log files and see if you've got any messages in any of those that gives an indication of why something is wrong. And if it's persistent, maybe after the end of this, I'll stay on a few minutes and other people can come and go and we'll try to figure out what's going on with your situation. Okay. So I can't really start the cleanup process. Oh, okay. I've got only 1 job remaining. Right? I've got number 6, my last guy. It's the only guy sorted. Number 5, 1 to 5 is done for me. So it's good to take a break. It's not an hour and 45 minutes in. We can sort of guess. We know the files are roughly all this. The number of reads in is about the same, similar to all these. So if we sort the BAM files by size, maybe to avoid confusion with the sorted ones, which we don't have yet for that guy. We'll just ask for the post BS-Bolt ones, not leave the sorted ones in there. And sort by size, that's the capital S to LS. We can kind of guess number 6 here is this F2, which should be very similar to the E1 version there. And it's at you know, that's 800 meg almost. It's at, but the other guy that's done is 836 meg. So we gotta be close. Right, so my number 6 is getting close. We can just estimate that from the file size. There's a guy, yeah, so he's getting close. Yeah, so it's important to note that just because a job disappears from the queue, it doesn't mean it executes successfully. It just means it's no longer running. And there can be lots of reasons for that. That's why we have all these log files at the job control level and at the analysis level. Done. Okay, I've only got my interactive session left, so I'm complete. So, okay, so let's look at the totality. Now I've got all my log files are complete, so I can look at all the E files, the standard error files. That's nothing new there. Those we can just delete because at this point, because don't delete yours yet if you're not done. If you delete them, you won't find out what would have gone there if you hadn't deleted them. It's not yet complete. So anyway, yeah. So these are fine. They don't carry really any information we're saving, and they've done their job, which is to tell me that this part of the job went okay. So I'm going to get rid of those. Actually, the PE ones are all, and the PE ones are all, the PEs and POs are all 0 lengths. They're empty too. So I'm going to get rid of all those. No sense in keeping those. That's the E's, the PEs, and the POs. All right, so those are gone from me now. And I've got only the standard outputs at the job control level left. And I'm going to use more instead of less, and I'll show you why. So if I do more with a bunch of files, it shows me the first one. It's a little different than less. It doesn't take over your screen as much. And it also gives me this little kind of header that says, this is the file I'm showing you now. And if I just hold down spacebar to go to the next file, oh, I get the next one. If I just hold down spacebar and let key repeat take it over, these are all very short files. And what it's done is sort of attach just the name of each file to the top, which is very convenient because now I can kind of inspect them all. And I see also which name each of them has and so on. And all they contain is, the logging we added to our script. Script started, script finished, started, finished, started, finished, started, finished, started, finished, started, finished. So now I know all of my scripts reached their endpoint. So those log files have also kind of done their job. We could keep them around, but because they contain the timing information, which may be useful for if you do new versions of this, right? So if I can look here, I can look at number 6. Here we know number 6 was started at 10:33:47, 10:34 say, and it finished at 10:45:41. Call it 10:46. So it basically ran from 34 to 46, which is what, 12 minutes? So that can be useful information in planning things, right? We can now extrapolate what other size files are likely to take and so on. We gave this thing an hour limit. Now you can sort of plan around there, or maybe increase the length of 2 hours. Anyway, you got to figure it out. It's like keeping log information like this allows you to sort of make some intelligent estimations when it comes to increasing the size of the jobs or doing big jobs and so on. Okay, so those log files we got investigated. I'm going to go into my stage 3 directory now and take a peek here what's going on here. Now, let's look at all these standard errors I got here from the analysis level standard errors. Nothing to worry about, right? Just some log information, progress information from SEM tool sort. I'm going to get rid of all those. Those aren't worth keeping. Again, if it's still in progress, don't delete them yet. Okay, so that gives me the standard outs, which I'm going to keep because those have at the end of them this stats from BS-Bolt, which is useful. Okay, so they're there. Now there's all these lines that come from BWA that we could get rid of. So, you know, one thing I could do is there's various things we could do, but you know, suppose I just want a quick overview of, say, mapability or something, right? So I could just cat all these together and grep out mapability. And now I see for my 6 guys what they were. Oh, they're all high. No problem. Okay, let's look at, you know, unfortunately, it doesn't give us a percent for mapped uniquely, right? Because it quotes it as number of ambiguous reads as an absolute number, which you have to divide by the total number of reads to get a percent. But so you have to know, you know, this varies for our different libraries. So we're not going to get a percent here, but you know, at worst, it's about 300,000 reads, and actually, you know, there's that 300,000 is kind of an outlier. There's 300,000, 160,000, and then 100,000. Let me be closer. Who's the 29? Who's this guy, right? So, and grep tells us it is 116604 F2. It's this number 6 is the guy that has kind of a more ambiguous ones than most, and we need to know the size to see if actually that's just because 6 has more reads than most. Yeah, so these are the number of reads versus number 6, right? So here's number 6. Now he has about the same number of reads as number 5, and it's pretty similar to actually number 2 as well. So yeah, actually, so somehow that number 2 has more ambiguous reads than kind of you know, whatever. So for whatever reason, but there you go. I mean, there's how to sort of deal with, you know, these kind of log files and keeping them around is useful and so on. So okay, so so that's great. Now we've got the BAM file. So everything looks good at this point. All these both, and then if I look at the BAM files, both sorted and unsorted, they're all of similar sizes, right? Actually, the sorted ones are a little smaller, which makes sense because once you organize the reads so that the alignments are all in kind of chromosome order, ones nearby each other are talking about the same sequence, which means the read-based calls which get embedded into the SAM file are all kind of the same near each other, which means that the gzip compression should do better. And you see that effect. So you see that the sorted ones are actually a little smaller than the unsorted ones, which is common. So that, and those sizes, you know, besides that, they're kind of comparable. It doesn't look like there's any gross values there. And we checked the standard errors and the standard outs, and we don't see any real error messages in there. So we're confident that actually probably that these sorted BAMs are good, or at least as good as the BAMs direct out of BS-Bolt are. And so I'm not going to keep the BS-Bolt BAMs anymore. So I'm going to delete the star. Now I don't want to delete star.bam. That's going to delete all the BAMs, including the sorted ones. That's no good. I got to make sure that my pattern here only pulls out the guys that are not sorted, right? So if I do 3.bam, that gets the BS-Bolt guys. It does not get the sorted guys, which are D.bam. So I'm going to get rid of those now. Okay, so I'm only keeping the sorted versions. And this looks good. This is the output, essentially, of this BS-Bolt alignment analysis. It's mixed in currently with the stage 3 inputs, so maybe now I'll clean up and I'll make a stage 5 directory and move those over to isolate them. So now stage 5, this is this is BS-Bolt align or whatever, you know. Let me just see what I call it just to be consistent with the slides here. So it's a BS-Bolt align. Okay, so BS-Bolt align. Okay, and then I'm going to move the stuff in stage 3, which is the BAMs and the standard outs. So it is the BAMs, the BAM.bys, and the standard outs. And that's what I want to move now into stage 5. So that's done. And if I look at my stage 3 directory now, that is back to the way it started. That's tidy. And now our new stage 5 guys are here. That is the sorted BAMs, their index file, and the standard outs. And since I did seem to keep the standard out information at the job control level, I am going to move those now into the stage 5 directory where they belong. And now my top level is also a little clean. Okay, so that is all good. So we talked about that. So why don't we take a look at what's in these BAM files now? Okay. So basically the job submission thing all ran well. The actual commands ran well. Now we can actually start to look at the data that we got out. All right, so we were able to successfully run it. Let's look at one of these BAM files. Okay, so what is a BAM file? As I say, it contains alignments. Well, okay, can we view it? Well, Les knows this is not looking like some text that I can really show you. So I said N there to not do it. Or if you want to look at it really, just say Y and you'll see it. Well, you can see it's kind of gibberish, which is all these bracket hex, two hex digits. These are individual binary bytes. Some of them are printable characters, which is why it doesn't look like anything human interpretable. It's not intended to be. Okay, so there is this file command that uses a whole bunch of heuristics to try to figure out what a files is by its contents and not by its file extensions. So we can ask what file text one of these things is. Oh, it looks like gzip. It's got some extra wackiness to it, but it's basically a gzip file. So I could unzip it on the fly and pipe it to less like we've done a few times and see if that helps. Well, that looks a little better. You can see there is some plain text embedded in the thing now. There's still some binary crap in there, and this is still clearly not intended for human consumption. And so that's because, it's not intended. BAMs are compressed, and they're not intended. They're binary. They're not intended to be viewed by humans. The SAM files, which were the predecessors to BAM files, were plain text and are intended for readable by humans and as well as tools. And the BAM files are really compressed versions of SAM files. So the question is, can we get a SAM file back up from the BAM file? And the answer is yes, and that's what SAM tools, one of the many things SAM tools can do. And it's what the SAM tools view subcommand does. And view is basically the Swiss Army knife converter. It can take SAM to BAM, BAM to SAM, SAM to CRAM, CRAM to SAM. It does all the combinations. So SAM tools view, and then you give it the name of the BAM file or whatnot. So I'm going to give it the name of one of these BAM files. This is the number 1. Okay, and then I'm going to pipe it to less because that's going to produce a lot of output. And this is what the plain text, this is what a SAM file looks like. This is the decompressed form of that BAM file. Now we're going to talk about all these columns here, but by default, the SAM tools does not show you what's called the header of the SAM file. If I had the dash little h there, I'll get the header too. So that added these little lines at the top, which is really the complete form of the SAM file that this BAM file represents. Without that .h, it just omits the header, which sometimes contains useful information, not particularly here. But anyway, that's some more metadata about the file. If you do the capital H instead of lowercase h, you get only the header lines. Okay, so that's the 3 flavors. But the default flavor is to not give the header. So this is basically the SAM file. Now, what's a SAM file? Well, okay, so there is documentation for this stuff. This is a very common file format in bioinformatics. If you say SAM file format in Google, it's going to take you to the sequence alignment map, which is what SAM means, file format specification. And here is the PDF file that defines this thing. And you can read it. It's long, but it sort of makes sense. It's as complicated as it needs to be. These are the definitions of the columns. We'll go through this for a second. And there's also a companion file to that file that somewhere, here we go. I got these links here. So, oops. So it's SAM tags. So if I go to the SAM tags version here, this is a companion file that the last column of the SAM file has all kinds of extra key value information. And there's a lot of different key value pairs. Some of them are well-defined, like in this file. Others are liner specific, BS-Seq specific. Anyway, we'll get to that in a second. So those are the documents that really define what these files are, but we're going to just I'll just give you a brief overview of what's in these without really going through all the details. This is some header metadata. We talked about that. Then it's really a tab-separated bunch of columns. And the lines are so long, it looks messy here. But this is really, if you had a really wide window, they would just be one line, one window line to each logical line. And there would be just a whole bunch of tab-separated columns. And here's column number 1, which is the read name. If you look back at our original FASTQ files, these are the read names, at least the first word of them, which is all that BS-Seq kept. So that's the read name. And the second column is this compacted form of a whole bunch of information. There's a whole bunch of yes or no's, binary bits about the alignments. The zeros are basically good alignments, primary alignments to the reference plus strand, and the 16s are basically good alignments to the primary reference minus strands. The 2048s and the 2064s are supplementary alignments to the plus and minus strand, which often you don't want. There is this website you can go to. Now that you can read the documentation files and you can figure out exactly what all those bit fields mean, but you can also go to like the BAM flags decoder or something like that. Explain SAM flags. Yeah, this is a little website somebody put together, and you can type in one of these decimalized bit field codes, and it will tell you which flags are set or not. And this says it's a supplementary alignment, and it's not, the read isn't reverse complemented, so that's on the reference plus strand anyway. So yeah, so there's all these flags, which mostly carry information about whether it's a primary or a non-primary alignment, and whether it's the plus or minus strand. A 4 would be an alignment, an unaligned read. At the bottom of these alignment files, you'll find the unaligned reads, which will have 4s in the flag place. The next column, column number 3, is the chromosome name in the reference genome to which this read aligns to. We only have one guy in ours, so they're all going to be 21s, which is the name of the chromosome in our reference genome file. Okay, so that is that. The next column is the position. We were talking earlier about the sorting thing using the one based quoted on the plus strand leftmost coordinate of any aligned base pair in the alignment. That is what's called the POS. That is column number 4 in a SAM BAM file. And here it is. All right, so these are coordinates telling us where the alignment is. So the pair of column 3 and 4 basically tells us where the alignment starts on the reference. This next column is what's called MapQ, which is very similar to the Fred scale used in per-base quality scores that we talked about at the FASTQ file stage. This is the confidence of the aligner, in our case, BS-Seq. The aligner's confidence that it's placed this read correctly on the genome. And it's in the same scale as Fred. So 0 actually is not at all, like not certain, basically best case is it's probably not where I decided it is. Usually because it's ambiguous. And once there's, like 10 places it could go, the chance I'm going to pick one randomly based on the weighted probabilities, with the chance that it's actually right is very low. So 0 is that. 10 would be 10%. 10 would be 90% certain of where it is. 20 would be 99% certain of where it is. By the time you get to MapQ 20, it's fairly certain where it is. But basically, things 0, 1, 2, 3, 4, 5, these are very not certain where it is. Now that we're looking at the file in some order, it turns out that the beginning of the file has only very low guys. If we open this up here and maybe scroll down a while, I'm sure at some point we can find some guy that's got some confidence there. We might be in a repetitive region of the genome or something here. This is a sorted file, so well, yeah, there they are, right? So here's a 60, right? So that's very, very certain. Here's a 34, pretty certain. And actually, all the ones on the screen right now are pretty good. This is what's called the cigar, next column. This is actually the details of the alignment, right? How many base pairs are lined up in an ungapped block, then in the gapped block, who's got the insert, who's got the deletion, how many bases, how many bases do I have to ignore at the beginning and end of the read that didn't align? All that stuff is in the cigar. It's a compactified format. But like this first guy, which is 41S23M, it's basically a number, which is a number of bases, and then a code that tells you what the interpretation of those bases is. So this is 41S bases followed by 23M bases. 41S is soft masking. That means the first 41 bases of the read didn't align. All right, skip the first 41 bases of the read. Then 23 bases of the read are participating in an ungapped alignment. And that doesn't mean that they're all exactly matching the reference. It just means it's an ungapped alignment, right? So matches and mismatches could be in there, but it's an ungapped block. So this 64 NT read is, ignore first 41 bases, pick the next 23. Well, that doesn't sound very good. And that's a big part of the reason why probably this guy has got a MapQ of 0, because it's only 23 bases that participate. The location specificity is not that big. Although this guy is a 64M, which means all bases are in a gapless block, but he's still very ambiguous. Anyway, looking here, actually, the most common thing we see is 64M, which is what you want to see, is an end-to-end alignment. It's an end-to-end gapless alignment, right? So that's about what you want to see. Let's scroll around. Let's see if we can find anything that's got some interesting stuff. Well, here's a 22S, 21M, 21S. So this one, the read has the first 22 bases and last 21 bases skipped, and the middle 21 bases are in ungapped alignment. And here's a guy with a deletion. 12 bases ungapped alignment followed by a 1 base deletion. And yeah, I have to look at the spec to remember whether that's a deletion of the read relative to the reference or deletion of the reference relative to the read. It's probably a deletion in the read relative to the reference, but an I would be the other one, whichever it is. Followed by 52 bases in an ungapped alignment. Anyway, that's all that kind of cigar stuff encodes. It's basically the details of the alignment as you would write it down in a little 2-line pairing or whatever, like you've seen in BLAST ports or whatever. Column 7, 8, and 9 are related to paired ends, and so they aren't relevant for us. Column 10 is a copy of the read base call sequence, ACGTNs. And if it's to the minus strand of the genome, this has already been reverse complemented. So this is basically the reads as if the read mapped to the plus strand of the genome, even if it didn't. This is the copy of the Fred's plus 33 per base quality scores for the read, also flipped in reverse order if the read aligns to the reference minus strand, so it's in sync with this previous column. And then the rest of it is column 12 to the end. And these are these key value pairs, which some of which are standard. There's many kinds of these. Some of them are aligner specific and so on. There's all kinds of stuff in there. The one that's most interesting for us is the XBZ. The Z means it's a string, and the XB is just the 2-letter code that it's been given. It's aligner specific, which is why the X is there as a sort of one of the no man's lands that anybody can, X, Y, and Z sort of is anybody can define what they are. The other ones are kind of reserved for the SAM-BAM people to define. And what BS-Bolt uses XBZ to communicate is a compressed form of which bases this read declares to be methylated. And so the upper and lower case is methylated or not methylated. The upper is methylated, lowercase is unmethylated. So that's basically the uppercase is a C, a read C aligning to a genome C, and the lowercase is a read T aligning to genome C. And the X, Y, and Z gives the context. So the X's are CG context, the Y's are the CHG context, and the Z's are the CHH context. And then these, it's kind of like cigar, where like the digits are sort of telling you copy of things. Anyway, so this notation, actually, I think I'm responsible for, because this is kind of the X, Y's, and Z's were how I communicated in the original nature work for BS-Seq about which sites are doing what. Okay, we want to visualize these things, right? We got alignments now. Let's look at them in a genome browser. Okay, so let's do that. We're going to need to download these things. How big are they? So the BAI files are super tiny, right? So that's 700 KB. That's 0 basically by modern standards. How about the BAM files though? 2.4 gig. Well, that's not crazy these days. I mean, if you talked to me 20 years ago that you could casually download 2.4 gig of data over your home internet connection, I'd be surprised, but it's pretty routine today. So we're going to need to download these. So how are you guys doing? How's your jobs doing? You got at least one finished, everybody? Or, I mean, you don't have to download them all 6. We can just one's good enough. Oh, it looks like you guys are mostly finished, and you got one of you that's only got your job number 1 launched. Okay, so if you've got at least one BAM, you're going to want to download that to your local computer and the associated BAM BY file. Okay, so I'm going to move to I need the other window where I've got my local machines terminal. Yeah, here's my local machines terminal. I'm going to go back up to my local machines desktop J2 directory. And I'm going to grab the BAM and BY files there. Okay, so you got to replace this with where your scratch space is. And if you haven't put them in the stage 5 directory yet, they're still in the stage 3. You can grab them out of stage 3. And if you just want to do one, you can just get the name here. If you want to do them all that you got, you can do this. But I'm going to grab them all, the BAM BYs and the BAMs. The BAM BYs first, very short. So anyway, so this is going to run. I just grabbed all the BAM BYs. They were that small. And now it's working on the first BAM. And I'm getting 8 megabytes, 9 megabytes a second on the home connection. That's not too bad. It's 100 megabit connection. Theoretical max should be around 12.5 megabytes a second. It's got to fight with the Zoom video and whatever the wife's doing and stuff, but we're doing okay. Time Warner's having not too bad day today. They don't always do well. If you're at school, you probably got a great connection, probably instant practically. But even on your home connection, probably is not too bad. So you can see we've already got one complete one. Over now number 2. Anyway, okay. All right, well, let's get on to IGV. Okay, so I've actually, you got to start. Oh, I don't know if I shared IGV in my screen share. Okay, so I might have to stop screen sharing. Maybe let me see. Yeah, there's no green border around the IGV window, so I did not include IGV in my screen share. So I got to stop screen sharing and share screen. And maybe for the moment, I will just do the slides and IGV. Well, I'm going to just do that. Slides, 2 keynotes, Firefox, and an IGV. Okay. We don't need Firefox right now. Okay, so here's IGV. Now, the first thing to do, this is already, I was playing around earlier, so you got yours will be blank right now. Okay, so when IGV starts, you get a window like this, and you're going to need, the first thing you're going to need to do is to specify the genome. Now, you may, in our case, you could use there's some genomes that are baked into IGV, and you could do that because our human GRCH3A is going to be in there. But I want to show you the more general case of dealing with a situation where your reference genome is not in IGV. So I got to navigate to where I've got these files downloaded. There's my day 2 directory on my desktop. Okay, and oh, oops. I only downloaded the BAMs and not the, I didn't know there's one just arrived. I didn't download the reference genome yet, which I need for IGV. So I also got to grab a copy of the reference genome. Okay. Well, because I've already got a download busy there. I've opened another terminal window on my local machine, which you can't see because it's not added to screen sharing. And I'm just going to run another SCP command. And then when the download of all the 6 is done, I'll copy this command into there just so you can see where I'm going to compose it while we're busy there. So we need our reference genome. There we go. Oh, where is it? We have the sequence already there. We don't have the gene annotation file yet. But, okay, back to Hoffman. What the heck did we call this thing? Okay. It is called, this is the name of the FOSTA file. What directory is it? So this is the path to where I got that thing. And so, I don't need my other window now because the download is finished. So I can do, that is the path on Hoffman. So I'll prefix the Hoffman degree use later use. Okay, so that's where I want to copy from, and I want to put it here. All right, so now I got a copy of the reference genome FOSTA file on my local machine. Unfortunately, IGV, even in the current version, cannot deal with a gzipped FOSTA file for the reference genome. So we've got to gunzip it. Okay, so it's expanded now. It is just a plain text file sitting here. And that's the way IGV needs it. We could show things right now, but it's useful to have at least a genes track and so on. So let's grab the gene annotations before we play with IGV. So let's close these. We don't need these SAM. We don't need these SAM things anymore. Okay, I'm not sure which. I think I just maybe killed the window, Firefox window that screen sharing had. So let me just show you the slides. But we need to grab the stop sharing again. Share screen. I lost all my Firefox. Two terminals. Firefox. Back to Firefox. So back to Ensembl. Human. And I pointed out the kind of the place that is to the right-hand half of the screen. Download GTF or GFF files for genes, blah, blah, blah. We will do the GFF3 version because I think they split them by chromosome. And so there is a chromosome, you know, there's the whole genome's annotation file, but it's big and we don't need the other chromosome. So here is the chromosome 21 GFF3 file. So I'll grab that. And I got the link. And I need to download that on my local machine. Well, we put it in Hoffman because it does belong with the reference genome file. We might as well keep them as a pair. So I am on my Hoffman place, and I will download with wget the GFF3 file there, and I will use my little trick to keep a log of how that goes. That'll be the file name. There we go. It's smaller than my machine. There it is. So it's on my Hoffman now. It's this BSeq file. I'm back to my local machine window. And here is the last copy command I did. And the name of this thing is this. So that's the file I need to put here. So now on my local machine, I have a copy of that file. I could have just downloaded it directly from my Firefox, but you know, I did the download in Hoffman where I keep it parallel with the genome file, and now I got a copy of the genome file and the GFF file on my local machine. The GFF file is a gzip file. It is in the GFF3 file format, which is yet another bioinformatics format. We can take a look at this file. We're not going to really, it's not really of primary relevance to us, so we're not going to really go into detail what's in here, but you can just kind of glance around and see. We've got, actually the first column is the chromosome name that the line belongs to. Then there's the annotator, the source, or the Havana is an internal project inside Ensembl that deals with gene annotation. So this is the source for where these gene models came from. There are different intervals of different types. This is the span of a whole gene. This is for an isoform. This is an exon. This is a little coding sequence chunk. 3' UTR, and there's all kinds of little regions. Anyway, then there's basically some coordinates that declare the one base start and end position of these things. So this is telling you the place on the genome where this is. This is probably a score or something which isn't relevant. This is the strand the interval is on, plus or minus. Then this is related to codon framing for CDSs, because for CDSs, they don't have to start and end on codon frames. So it needs to be a little way of telling you exactly sort of how the codon framing is relative to the interval. And then there's a bunch of key value pairs, like every other bioinformatics format. And Ensembl, this is a very ensemble-y looking gene key value pair stuff. And you can kind of see there's some useful information here. Like this is the systematic name of this gene, the Ensembl systematic name. This is more the human name. It's a protein coding gene, because not all genes are protein coding. There's non-coding genes, there's RNA genes, so on. There's all kinds of types. And then this is some free text human short name for what this gene is. And wow, actually, this is really a complete coincidence, but it's very fortuitous that this gene's name is a novel protein similar to a cytosine 5-methyltransferase. It is DNMT3L. That is, DNMT3 would be one of the methyltransferases that puts the 5-methyl mark on stuff, which is just a complete coincidence. This is one of the homologous things to that, so it's a methyltransferase 3-like. But anyway, what a coincidence that that's the first one on chromosome 21 in the annotation file. Anyway, so that's what that looks like, and we can keep it gzip. IGV is perfectly happy to use a gzip GFF3 file. It's just that the genome files doesn't particularly like to be gzip. Okay, so now we are back on IGV, and for some reason the Zoom green bar that tells you that this window is shared is not around this window. So tell me that you can see the IGV window. Yes, it doesn't look to me like you can see it. I thought I shared it, but maybe you can't. Do you see the IGV window? Yeah, you do. Okay, well, whatever. The green border doesn't seem to be too robust. Okay, so yeah, so we're ready to. Okay, so you probably got a blank screen. You go to genomes, load genome from file, and you point it at that .fa file that we did. Okay. And so we have now loaded that as our reference genome. There's only one chromosome in there, so this box doesn't look too exciting. Normally this would be the place you pop up different, this is where you pick the genome if you've got multiple genomes installed. This is where you pick the chromosome you're looking at. But we've only got one chromosome, so not very exciting. And yeah, we're looking at the whole 46 meg of chromosome 21, but we have no tracks loaded, so very, very, very dull. Let's load the, we don't want to be here anymore. Now in file, load from file, we can start loading data. And so, this directory is wrong. So I got to go to my desktop, day 2, and actually you can see there's a little FA file. This is an index FASTA file. So the FA file is a random access speedup for FASTA files, which is something IGB just made. It does have some intelligence built into it. So here's the gff3.gz file. That is the track I want to add. Open that up. And IGB is thinking. And well, we're actually showing the whole chromosome, so that's all the genes. Okay, so I got to make this window a little bigger because obviously it wasn't designed to be that small because this little zoom thing wasn't even showing all the way. So this little plus sign means we can zoom in. You can see right now we got a span of 46 meg in the window. Now it's 31 meg, 15 meg, 8K, 4 meg, 2 meg. Actually, we're starting to get to the point where individual genes are showing. There's 1 meg, 500 KB, and now we're at 250 KB. You can drag it. If you click in the white space and click, you can use your mouse to kind of scroll around. I think the arrow keys also work. Yeah, they also work. Not too exciting because we got the gene annotations track loaded, but that's something, right? So here we can start to see some gene names, and so there's the structure of them. These guys with the -201 is an isoform. This guy without that is probably the span of the gene. There's different interval types in there. We're seeing all the interval types that were in this file, not just the gene structure ones. Anyway, yeah. So there's here's a long intergenic non-coding, right? So there's link RNAs, whatever. I mean, I'm just looking for anything with a good gene name. MRPL39, that sounds pretty normal. JAM2, that sounds really normal. Okay, so there's some genes. We don't have any our data loaded yet though. So load from file and feed it some of these BAMs. Okay, so let's feed it some of these sorted BAMs. All right, and it needs the associated BIs, but they're already here. Okay, so I'll just load up all of them. Why not? Let's get the 6. All right, so I just fed it all 6 BAMs. That's command click on Mac or shift click maybe on try. You can control option command or shift. One of those will be your way to select multiple. Okay, we'll make this window bigger. Oh, let's see some alignment starting to show up. So I'm going to zoom in a little closer because we're still pretty big. Okay, so that's 82 KB. Actually, this track that just showed up is the nucleotide sequence starting to show up. The colors are their color code for the 4 bases. I'm trying to aim at the promoter of something. So these are these 2 genes in this region here. This is a minus strand gene. The arrows point that way. This is a plus strand gene. The arrows point this way. Somewhere in here will be the promoters of those 2. Well, actually, this is 40 KB, so the promoters will just be really close here. I'm going to aim in the middle, but we might have to scroll around. Okay, so 40 KB window, 20 KB window, 10 KB window, 5 KB window, 2.5 KB, 1 KB window. All right, let's try here. Gene tracks is interesting. I aimed in the middle. But let's see if we can scroll down somewhere and find our reads. Come on. Come on. It seems to have decided to use enormous amounts of vertical space to show these, but we can start to see we got reads piling up here. All right, here's a little pile. Let's zoom in closer. 300 bases, right? The window span left to right is only 300 bases now. So you are actually starting to see individual bases now, 162 bases. If I scroll back up to the top, well, let's we got to go even closer. 80 bases. Now you can see the individual letters. T is red, A is green, C is blue, G is yellow. All right, now we're showing the plus strand. I think if you click this little arrow, now we're showing the minus strand. Okay, there's a little strand arrow. Let's go find some of our reads, okay? Now I've got these turned on in like super compactified form. If you right click, there's all kinds of options for how you can display your business. So if I go from squished to expanded, you'll see the actual reads as kind of reedy looking things here. So these are the actual bases of the reads. It'd be nice if we could get one of these close enough to the sequence at the top that you could see them. Right now, I don't know why my gene annotation track is kind of sucking up all this space. Maybe I can move him above. and get him out of the way for now. All right, there we go. So at least now I can get the reference sequence adjacent to the pileup. So you can see that basically that, the reads basically are calling the reference very well. The aligners done well, the reads are sequenced well, and so on. So all that looks great. We'd be interested in methylation though, right? Well, okay, here's a genomic C with some reads calling T and some reads calling C. Well, that's good. It'd be nice if we could see that in a less insane way. All right, so if you right click, here's some settings I'm going to recommend. So group alignments by strand. That'll turn each track into essentially 2 mini tracks where the plus strand alignments are on one and the minus strand alignments on the other, which is kind of important for bisulfite because they're not really, the one C's are on one strand, the other C's on the other strand. So, okay, so we'll do that. That groups them kind of that way. Color alignments by, and this is very important, bisulfite mode. And I will say none. And now what you see is red and blue indicating, the reads have all been grayed out, except where we have C's and T's in the reads on top of genomic C's. So the methylation information from the bisulfite assay. And if I hover over this one, it tells me I'm hovering over a base C in a read. If I hover over this one, it's telling me I'm hovering over base T in a read. So the blue ones are read T's and the red ones are read C's. Okay, so the red ones are methylation and the blue ones are unmethylated. And this is a CCA, so that's actually a CHH context for that C there. Next one is a CHG, so this one's in CHG context. Let's find some CGs. So this is a CG, right? This one is a CG, right? And that is fully methylated. What about its mate on the minus strand? Because this G mates with a C and the minus strand is down here. So if we scroll down to find the minus strand reads for that guy, well, guess what? They're all basically fully methylated too, except for 2 reads that are calling unmethylated for him. So that pair is pretty much consistent, and our coverage is pretty good on that guy. So we're pretty confident of the methylation percent on that guy. All right, now you kind of get it. Let us go to squished. We don't really need to see. That'll just pack each line into each read into a single pixel, right? So now we don't really need to see the individual guys to that detail. Now we kind of get our little pileups, right? And then the tracks take up less vertical space, although I really would not like it to use as much white space as it's using. I don't know why it's in that right now. So the next thing to do is since this is human data, and we're mostly concerned with CGs, we don't really need this blue-red highlighting at every genomic C. We kind of maybe just want to see the highlighting at the genomic CGs. So then you can go to color alignments by sulfite mode and just pick CG. And now only the CG positions will show with the blue-red coloring. So these are all CGs now. Now that is now a symmetric pair. You see that. So those are our 2 little guys that were blue before. Just it's on squish mode now, and that's a pair. Okay, that's a pair, and so on. All right. So now we can. I don't need my C's to be this wide anymore, right? So let me zoom out until the C's are roughly a pixel across. Okay. So now we can fit about a 1.2 KB. And we're scrolling on. You can see there's a lot of reasons we don't have coverage, okay? And then we get little patches where we have coverage and so on. And okay, there's some methylated CGs. We're kind of in between 2 genes right now, so methylated CG is not really a surprise. Here's an unmethylated one. We don't have coverage on the minus strand there, so plus strand has coverage. Here's a bunch of reads with coverage, but they don't cover any genomic CPGs, so not helping us learn any methylation there. That's coverage, but not at places we're interested in. Here's more of a mixed site, right? Some red and blue. Okay. All right. Well, now that you kind of know how this goes, so let me zoom in a lot. I'll zoom out a lot, right? So actually, I was able to get to about 40 KB and still kind of see what's going on. And now we can start to get a little better idea of what's going on. Well, there is a little unmethylated patch. Right, so right there, and it's C methylation. So that's a little patch. Here is another little patch, okay, of unmethylation. Now, somewhere, if I scroll enough here, I thought we were going to see it. Oh, I lost track of my gene track. So here we are. All right, so the genes are up here. I've got this would be, well, this one is a minus strander. This guy's a minus strander. This is actually the 3' end of both of those genes. And actually, there is the unmethylated patch. I'll center the window on that. Okay, so this unmethylated patch, which is now roughly in the center of the window, is actually right upstream of this MER 155HG. So that's probably a CG island associated with the promoter of that gene, and that is unmethylated. I mean, it really looks like quite right. I won't move my mouse horizontally, and I'm just going to scroll height. There we are. It's right in the right place. Yeah, I'd love to have it repack. It should just auto do that. Fit data to window. Even that. Well, that's a little better. Still. All right, we may have to manually specify our track heights. It only says 40. That doesn't look like 40. Yeah, I don't know. It's being resistant about hacking these. Actually, this version of IGV I have installed seems to be a little unstable, at least on this machine. But anyway, clearly it works, right? So it's clearly working. All right, you can hopefully get a view yourself going. All right, these other tracks, I was hoping to see whether that's a differentially methylated site, but apparently their coverage is such that it's on that we're not seeing the packup for. Because we didn't change the rest of these guys to bisulfite mode yet, right? So I didn't kick all of these into CG mode yet. So let me quickly kick them all into CG mode. Okay, bang. Well, that's clearly unmethylated in that sample. Okay, how about this one? Same thing. Okay. Oh, whoops, lost it. That's 3 of them done. Okay, there's another one. All right. I mean, I know some places to look at. So if we don't find something good in a minute, we'll just go to a known place. Okay, nope, doesn't look differentially methylated, right? So it's that little promoter. Well, maybe the extent of it, we'd have to check the horizontal extension system, but that little, presumably CPG island in this gene's promoter looks like in all the samples is unmethylated. Yeah, that's going to be the generic case. Most sites are not going to be differentially methylated. Okay, so let's see if we can we can get out to about 40 kb before we kind of lose all detail. So all right, so I'm just going to start clicking randomly. Right, here's the whole chromosome in this little box at the top. If I just click randomly somewhere, I'm going somewhere else, and things are going to load. All right, I'm just looking around. Didn't catch any obvious ones there. I'm going to click another random place. Maybe I'll try 2 or 3. If this fails, then I'll have to go look at the place I put in the slides, which I found after just a couple minutes before. Oh, there's a place with high coverage. There's some anomalous coverage, right? Bang. Maybe that's what's driving the track heights crazy. We might have to filter out some of the high coverage sites. Yeah, those are using the full vertical space. Okay, let me go to the site I picked out just by browsing around in the notes. So it is 21. Let's see. IGV is sort of annoying. It doesn't let you, if you switch out of its window, I think at least it changes the highlight, but it lets you keep the partial type stuff. So it's 1, 4, 2, 3, 7, 3, 3 to, then IGV's GUI could use some refinements, right? It's a little rough. Okay. 14, 2, 3, 9, 6, 9. Now, okay. This was a place I found with just a couple minutes of browsing on my own. What do we got? We are at the promoter of something. We can't see the name right now. Maybe if we hover over it, we can see the name. That's a link RNA anyway. All right, how about this one? Oh, it's ABCC13. It's marked as a pseudogene, but anyway, if it was a real gene, then this is where the promoter would be. So let's look at our samples. Let's go back to the bottom. All right, what do we see? Well, okay, this one, unmethylated. So, am I not seeing the patch? Oh, it's to the left, I think. It's to the left. It's this little patch to the left over here. Right, so, okay, in this sample, this little patch, red, right, methylated. Red, a little blue, mostly red. Red, right? We got 2 bioreps each. Well, we got 3 tissues, 2 bioreps each. Well, that is not red. That is much more half-half, right? That is much more differential. I mean, that's different, sort of. The extent of this blueness is much higher in these samples. The SRX 116604, which I got to look at the key in day 1 slides that'll tell you which tissue that is. So that's differential, and it's pretty close to this what's marked as ABCC13, which if I got a Firefox that's still alive, I think this Firefox window you can see. So if you just start searching ABCC13 DNA methylation, Google knows everything, right? So okay, yeah, some generic info pages about these guys, but then we get to papers, right? Let's look at some of these papers, right? So I'm opening these in tabs. Let's see. Expression related to hematopoiesis, and we are in a bunch of experiments that are with differentiated stem cells. One of which leaves the hematopoietic lineage. Okay, so that wasn't the paper I thought was most interesting. There's a heterochromosome euchromosome frontier at this point in chromosome 21. So heterochromosome usually gets heavily DNA methylated and so on. So is this paper. So here, this paper, there's this ICF syndrome, facial anomalies, centromeric instability, facial anomalies. And this gene, whatever it was, I should just search the name. Okay, 3 genes map to the duplication function heterochromatic domain, and 4 genes map to the duplication for euchromatic gene. Okay, and actually they studied DNA methylation at this locus, ABCC13, and in controls, it's like a third methylated, but in this type 2 ICF disease, it is 14% methylated. So now this sounds, very, we possibly have actually seen some real biology here. So, and this was just, as I say, browsing around for a couple minutes. So the thing we want to do now is automate this analysis and sort of find differentially methylated regions and so on. And that's what we'll do on day 3 with Methylation. But anyway, okay, so this hopefully gives you some flavor for what you can do in IGV. You can look at the details of individual reads, so you can check out basic stuff like, does your data have any problems and so on. And you can even use it a little bit for discovery, right? And you can certainly use it for checking out places that the automated analyses find, right? So you could look for like read disagreements with the reference or bisulfite conversion. You can look at the different contexts, CHHG, CHG, CHH, CG, and in human, you don't want to see much of the CHG and CHH, and you want to look and see genomically where that is. Am I finding in mammals my promoters unmethylated, in the CPG islands and so on. You can check for the CG symmetry. You can check for the coverage unevenness. You can check your fragment links if you have paired-end data. You can look for possible non-conversion. You can look for how much it looks like PCR-induced duplicates are a problem and so on. So you can check all this kind of, the IGV is a great way to check out all that kind of stuff and just kind of, play around and check out once you have automated methods pulling out intervals, you can check out the intervals in the browser. So that's a great tool that you're going to want to use when you do bisulfite experiments. Okay, so we are, in terms of the pipeline, we are now covered all this stuff, and we are now at this point. So the last thing to do today is the BS quantification. Okay, so that is the automating the little pile-up analysis on each genomic C and counting how many reads count C and T, and putting that in a text file in a way that downstream programs can use easily. Because the alignment file has got way more detail than you need for that kind of stuff. So that's what we do now. Remember from day one slides, this is kind of the generic situation, right? Here's the reference. Here's one of the strands of the reference. It's got a cytosine. And so we got reads piling up there, and some of our bisulfite reads have a T, and some of them have a C. And so the C's indicate, they were in methylated observation, the T's were unmethylated observation, the way bisulfite works. And so our best fraction of methylation at this particular locus is 2 out of 6, right, 1/3, right? So that's somewhere we want a text file now that says on this chromosome, on the plus strand, at this coordinate, you got 2 yeses and out of 6 total observations, right? Essentially, that's the data we want, and it's what we need, for example, to start the DMR column and so on. So that is the job that we need to do next. And BS pilot, that task can be done with BS-Bolt, no problem. It's called call methylation in BS-Bolt. And this is the command line options for this. And now you just treat this the same way we did the alignment. Okay, we look at this help text. We find out that probably the options we want to pay attention to are these. The rest are either okay or defaults or don't apply. And it's basically you need to put the BAM file as an input. We need the BS-Bolt index again, what base name you want to give to the outputs, how many threads you want the computation to use, and then some quality parameters like how certain of the map queue, how certain do you want the alignment certainty, the map queue embedded in the alignment spam file, do you want that to be before you accept the read for tallying up the C's and T's. So this would be 20 as the default threshold. That would be 99% certain before you accept it. BQ is the per base quality that the individual read C's and T's have to be before you count them. This is set by default at 10, which would be 90% certain from the Illumina base caller that C or T is called correctly. And then there's 2 more parameters that are coverage thresholds. So what is the minimum depth of coverage before you want the cytosine to be in the output file? And what is the maximum depth of coverage? What do you want the cytosine to appear in the output file? Why would you have the minimum coverage? Since we can do the filtering later at the aggregation stage, we might as well set the minimum to 1 to keep all the data. We can always filter that later. Why limit the maximum? Well, you already saw that one weird place where like a huge number of reads piled up. And the reason why maximum coverage is something you want to filter is because the repetitive regions in the true genome are not generally fully correctly represented in the reference genome. I mean, one hint that that's going to be true is that there's 5% of the reference genome that's gaps, ends, right? So the sizes of those are estimated based on various methods, and that represents a sequence we don't know that's not represented in the reference, and it's generally the hard to assemble sequence. It's the most repetitive sequence and so on. So what happens is you get reads from all over the true genome at these repetitive regions, these highly repetitive regions. But our reference genome only has smaller numbers, 1 or a few places that actually has representation for these homologous, for regions homologous to those. And so during the alignment process, it looks like that's the only place you can put them. And so you got some locus that's homologous to another bunch of loci, but you don't have all the homologous copies represented. And so in the true genome, your reads that are coming from these various true places are all going to pile on that one place where you have representation. And so that's how you get this sort of anomalous coverage there. And then the data that there is some average over all the homologous loci and not just the data from that one represented locus. And you don't really, you got to decide kind of what point you're going to call it odd, right? So it's hard of our a priori kind of hard to decide, and depends on the genome a lot, the organism a lot, what's appropriate. So one thing to do is you can do an exploratory run where you just set the threshold to basically infinity, 999 million or something, right? And then you can histogram the coverages you get out of that data in R or whatever. And then, there'll be some, most sites will be in some, range of coverage. We know our average is supposed to be something like 3 or 4 or 5, or depends whether you combine the 2 CGs and symmetric sites or not, or whatever. But you can do the histogram. You can kind of see where the meat of the data lies, where most generic cytosine fly. And then you can pick something to the right of the bump where you want to decide that beyond there is sort of odd. You're not going to lose many sites, and the tail of that will go way out into the thousands or whatever of coverage, even though your mean is like 5. And those are suspect places for the reason I mentioned. So anyway, yeah, it doesn't sort of matter for us what it is. Like if you do an exploratory analysis, set it to something high. Once you kind of see what the data is telling you is a reasonable threshold, set it back down to a lower threshold. Then we construct, work, and submit scripts just like we did for the alignment, and their structure looks pretty much exactly the same. Let me make those permissions set available to you, because I don't think you're going to probably want to do this on your own, because we're kind of running out of time today. We're coming up on 12 o'clock. Let me go to my Hoffman terminal window. Say where I am. Go to workshop stuff. Let me add the permissions for you to look at that stage 5 to 6 scripts. Now you've got permissions for all the stuff there. And you can copy that stage 5 to 6 CGMap BS both launch and work scripts to your working place now. So you've got those in there. And since they're already set up, ready to go, it's basically set up the same way the other one was. So if you run stage 5 to 6 launcher, it gives you the help again. Now instead of expecting fast QGZs as input, it wants the BAMs as input. And I've already moved my BAMs into my stage 5 directory, right? So if I look at my stage 5, I've got all my sorted BAMs in here, right? I've got 6 of them, and those are the ones I want to give to the BS-Bolt CGMap or call methylation stage, which is what we're talking about right now. So since the script's already sort of ready to go, and I can submit that job, and that can run while we're talking, right? So I've got the job submitted. I got 6 of them waiting. None of them have started yet. Oh, I got 3. So that can work while we're here. And yeah, so the submit script is very, very, very similar to what we had before. Very little changes. The work script is also mostly the same. I mean, the way it works as a flow is basically the outline you've already seen. It's exactly the same. We're going to embed some of the parameters into the file names. That's useful as breadcrumbs when you send your collaborators 27 versions, and then they come back 2 years later and they say, I need the methods for this. And you tell them, well, what did you use? And they don't know what parameters you use, but they say, I use this file. And at least that file name will tell you all you need to know about which version of parameters they use. This is basically the planned command. We looked at kind of the BS-Bolt help and how the call installation works. You need the database, the input BAM, the output base file, and how many cores you want to use, and then those parameters that control like coverage thresholds and the map queue, base queue thresholds. So that's basically that. The output of BS-Bolt call methylation is what's called a CGMap file, which is gzip, and it's a plain text file. It basically has the information we just talked about: chromosome name, genomic position, which context it's in. Is it a CG, CHG, or CHH, or is it the plus or minus strand we're talking about? And the fraction methylation as a point estimate, which is just done by numerator over denominator, numerator being number of reads with calling methylation, and the denominator being number of reads calling methylation plus unmethylation, basically usable coverage. And the thing is that the BS-Bolt file mixes in all the context, CG, CHG, and CHH together. I generally find it most useful to have that data separated out into separate files. So you have a file for the CGs, a file for the CHGs, a file for the CHHs. And there's a very small number of sites where you can't tell from the reference sequence because of the ends, or they're near the address of what they are. So you've got the rest of them, which is the unknowns, context unknown. And so the rest of the script, which is kind of this messy bit here, is to basically, after the BS-Bolt call methylation is done and it made the CGMap file, is to separate that file out from being mixed context to being a single, just separate files, one for each context. And we do that by just unzipping it on the fly and then piping it into awk, which is maybe something that you've seen in the UNIX workshop, which is a way to, it's like a little mini scripting language like Perl or Python. It's just very good for very small tasks like this, where we can essentially give the program as one of the command line arguments if the programs are so short. So here we're telling awk this is a tab separated file, so that -f :/t colon is to tell awk it's a tab separated file. And when awk talks about fields in the file, they're tab separated fields, as opposed to just white space separated fields or comma separated fields or whatever other options you might want. And then we look at column 4, which happens to be the column in CGMap files where the context is declared, and it's either CG, CHG, or CHH, or something else, question mark, I forget what, for the unknowns. And then if it matches that target context that we are for generating the particular file we're generating, then we print out the line. That printf %s \n$ just prints out the whole line. The $0 refers to the whole line, whereas $4 refers to the fourth column. And then we gzip the outputs, and then we put those into the individual files. And this T stuff with this with the right hand arrow, which kind of looks like redirection, which it is, and then the parentheses. This T in that construct is a way for us to sort of do all four files that we're trying to generate from the same pass with gzip. So we don't have to do. We could write this as several commands in a row and just do each file with its own pass through gzip. But this T construction allows us to, with a single pass through gzip, just sort of distribute the output of gzip to all of these pipelines at the same time. And we end up running this whole separation process in parallel. So we've got 4 gzips we're running, we got 4 awks running, and 1 gzip running, and all those commands, which is roughly 4, 8, 9 processes will all run in parallel, which is nice. So that's some sophistication, and then okay, yeah. So otherwise, it works exactly the same. So you kind of see how I like to do job solution on Hoffman. Now I got 4 of these running. I got all the same kind of progress files that I had with the alignment stage. The outputs are accumulating in the stage 5 place because that's where the inputs are. You see I've got this CG map file, GZ, which is being created. That's being created by BS-Bolt. None of these has finished BS-Bolt yet, so the splitting stage into separate files by context hasn't happened yet because none of them have gotten to that point yet. Clearly 4 of them have started now, and 2 of them are waiting. So you kind of know how that goes now. And well, okay, so so otherwise it works the same. We're going to clean them up the same. We're going to check for the errors the same way that we did with the alignment stage. This is, please, this afternoon you can play with this and see if you can get this successfully complete on your own. I do have some CG maps here that are starting to be ready, so we can start to look at these, even though they're not complete. You can less a gzip file, even if it's still being constructed. You can gzip CD it and pipe it to something, even though it's still being constructed. All that's no problem. When you reach the end, gzip may complain and it may be truncated or something, but it's fine to, we're just looking to see what's been done so far. So yeah, we can do that. This is what a CG map file looks like. It has exactly the columns that I was talking about before. We got the first column is the chromosome name. The second column is this genomic strand that the cytosine is on. Like a lot of bioinformatics files, everything's quoted on the plus strand. So the C's means plus strand and the G's are really the minus strand. They're all talking about genomic cytosines, but everything's phrased relative to the plus strand. So it's at where the plus strand has a G, that's where the minus strand has a C. So that's the strand indicator essentially in these files. They could have just as well said plus or minus. This is the coordinate for the genomic cytosine quoted on, well, I'll let it for you. Try to figure out on yourself, is this number counting from 1? Is this number counting from 0? How do minus strands work? And so on. With, say, IGV, you should be able to figure that out. You should be able to kind of zoom in close enough to some place and just work out an example and just figure out what the coordinate system in the CG map file must be. The fourth column is the context, which, since this is the map out of BS-Bolt, it hasn't been separated yet. Let me see if, nope, nobody's gotten ready for the separation point. So yeah, so the file's all mixed at this point. Okay? The little rest of the script that does the separation hasn't done yet. This next column is related to subcontext, like CHH, what that H is, that first H, that's what this is. You really need the third base too, which is, this isn't a complete subcontext, but this is sort of subcontext information. Of course, CGs, there's no subcontext, it's just always CG. The next one is the fraction methylation. Maybe when you scroll down to a place where maybe we get some not 0 and 1. This place is okay. So here are here's a CG site, 2 CG sites. Of course, they come in pairs, CG sites. And in the first one, which is the plus strand one, we observed 5 methylated reads out of a total of 7 coverage reads, right? So the estimated fraction methylation then is 5/7, which is 71% or whatever. This is, and then the third, you just take the numerator, this is the numerator and denominator, and then this column is just for convenience and gives you just the, the division as a fraction from 0 to 1, not as a percent. As a percent, you have to multiply by 100, right? So this is 5 out of 7, and then the other side of it is 4 out of 7. Okay, so that's how this works. That's you see, you know, that's it. That's all a CG map file is. And think because we have the coverage 1 cutoff, probably the cytosine sites in the genome that just have no coverage in the sample will not appear as lines in this file. If we had set a higher coverage threshold than the 1 to BS-Bolt, then we would get less lines in the file. Oh, finally, okay, we got one of them that's reached kind of the separation state. So now you see this is the CG map file straight out of BS-Bolt. And then you can see our separator is busy working, turning that into a CG underscore. I need to do something to kind of name it differently than the one from that BS-Bolt makes. And since the rest CH and CHH are 3 letters, it makes sense to sort of pad this CG to 3 letters. So the presence of that underscore tells me that that's a CG only file as opposed to CG map, which is probably a mixed file. Right, so if I look at this CG underscore map for, oh, they've been started, but they're not big yet, right? Those are all still empty. So somehow the buffering hasn't sort of gotten, oh, okay, there we go. Some time went by and now they have some non-zero size. So, okay, so this CG underscore map gzip file I can look at. And you will now see that this only contains CG sites. And it should be just a subset of sites from the BS-Bolt file in the order whatever BS-Bolt. And if I go here and I go instead look at the CHG file, you'll see that these are only the CHG sites. And if I look at the CHH map file, those are only the CHH sites. Okay. You can already kind of get an idea that like, oh, so CHH methylation is pretty low, right? You know, even when it's here, it's like 1 out of 12, you know, 1 out of 6. And this is basically going to be related to base call error and non-conversion that didn't get pulled out by the non-conversion filters inside BS-Bolt. So yeah, so this is 1 out of 3. Well, it's a low denominator. So you know, you're going to have these errors once in a while, and if it happens to be at a place where you have a low denominator, then of course the estimated fraction is going to be high. I mean, once you've picked a place that's non-zero, you know, you've already kind of selected out all the places that are all 0. So the average is still quite low, even if that individual site says 2/3 now. But it just got 2 copies that came out as high. The denominator is 3, so the confidence is really quite low, but it's really 2/3. But you know, he's surrounded by all these 0 over n's, right? Which is really the more of the picture with CHH. So there we go. If we look at CHG in human here, maybe we'll find some more that are a little higher. You know, there's some 1s out of blah once in a while. It doesn't even look that much different than the CHH stuff, which is what you expect. As opposed to the CG sites where 5 out of 6, 8 out of 9, 6 out of 7. Looks like a little high. Who knows what? Okay, you hit enough random keys in Unix and stuff starts to happen. Okay, so you know, here's 11 out of 13, 14 out of 18, 24 out of 27. You know, anyway, you know, obviously, and yeah, you kind of get a sense of the binary nature too, right? So generically, sites will be methylated, right? That the genome-wide average is whatever, like 80% or whatever. But we don't know that yet. We haven't looked at the. We can look at these output files here. So here's the standard output file that came out of here. This is the standard output file that came out of BS-Bolt quantify. And if we look at that one, you can see that BS-Bolt produces a very tiny little summary that actually has some key information. I saw this many methylated CpG cytosines. I observed this many, so the fraction of CpG cytosines that were methylated was 84%. And that's probably, you know, that depends kind of on how that's averaged, but that's you know, that's kind of, this is pretty variable, but this is in the range of what you expect. Much more important though is the CH for human, which, you know, the CHG, CHH, that should be very low in human. And it's 0.6% on average here, which is about what you expect from the non-conversion. I think the original paper for this said something like 0.5% or something. So we're kind of right in line with that. Anyway, so that's how it goes. So we are going to have this file ready for day 3. These 6 files. Well, it's really 6 times 3. Well, the non-CGs aren't really relevant for this stuff because this is a human sample. So we will only do the stat stuff on the CP sites. So we get 1 CG map, CG underscore map file for each sample. We have 6 samples. So we'll have 6 files that are relevant to day 3 where we start the the DMR stuff, and then we can do some functional stuff, functional annotation if we have some DMRs and stuff. So maybe we can have the statistics find us some better places, the best places that these sample have methylation, differential methylation relative to chromosome 21, right? It's really, you want to look at the whole genome, but asking for the chromosome 21, it's only like a 60th of the whole genome or something. So we may not have much, but I took a sneak peek. I think we'll have we'll have one or two sites that are really quite good. Okay, so you've got the you can play with this. We didn't really go through it to the detail that we did the alignment stuff. I think we covered most of this here in those last few minutes. I wasn't really syncing with the slides, but you got the slides. You can play around with it on Hoffman, and so try to get to the point where we'll be meeting for tomorrow. Okay, so I'll take any questions, or did everybody get at least some jobs started and so on? So you have a proof of concept, and you may have to wait until after noon here to get your data. But everybody got jobs started and at least some jobs finished. Or? Okay. All right, so any questions you want to ask? Or is this generally helpful? Is this good? These 2 days good?
Hi, Sean. Can I ask a question?
Yeah.
Right. So obviously, you're for teaching purposes, you've broken this down into a single chromosome, and therefore that affects like how quickly everything will be running. So I'm obviously asking from a selfish point of view that I very shortly will be coming into possession of a lot of data for mammals, mouse, and I'm just wondering what's the best way in order to start sort of trying to analyze that. Would you try to like do 1 or 2 files at a time, play around with how much memory and cores, etc., to be requesting? I mean,
right. So as a rough guess, right, chromosome 21, I think is about a 60th of the human whole genome. So you can take, some of these jobs were taking me, like 10, 15 minutes or something.
So multiply by 60.
Yeah, so multiply by 60. Well, certainly a 1 hour allowance is not going to work, right, then. You could try increasing the number of cores, up to, 4, 6, 8 or something. Of course, beyond 8, availability of machines starts to really tail off. And at some point, files get so big that you may have to split an input FOSS-Q file because you want to run it in parallel on multiple nodes, right? We don't really, like, BSBolt doesn't do that, right? BSBolt will use multiple cores on a single node, but it doesn't know how to like talk to multiple nodes at the same time and aggregate a huge number of cores by spreading its load over multiple machines. The way to do that is to break the input file up into smaller pieces, run the pieces in parallel, and then put the pieces back together at the end. You merge the BAM files. It's essentially concatenation, but you need to use SAM tools or something to do it because you can't literally just concatenate the binary files. They have to be processed. They have to be encoded right. So that's one way to do it. And at some point, files, maybe even by 60, maybe that is a problem. It probably was 60. One real big threshold on Hoffman is a day. Like if you cross, if you ask for a 23-hour long allowance, that's generally no problem and not really much different than the 1 hour, or 2 hours, or 3 hours that we've been kind of asking for, or our interactive sessions were 6 hours a day. But beyond 24 hours, it triggers like a whole different set of rules and stuff. And so you try to get things to have an allowance of under a day, and a day is 24 times longer than an hour. And some of our things were finishing in like 15 minutes, so you get about 4 of those in an hour. So 24 times 4 is roughly kind of almost 100, right? Which is bigger than 60. So it could work, right? Even with the 3, we've been asking for 3 cores in the machine, but if you scale up to you let the thing run a day, it might finish a 60x larger file. Of course, your coverage is on a modern platform probably, so you might be aiming for higher coverage than these were, which is, this is like 3, 4, 5, 6x, right? You might have 10 or 15 or something. So there's another factor too that might take you over the day limit, right? Well, but then you can increase the number of cores, right? So you could mush that up to 6, and there you get a factor of 2 back, right? So Maybe you could do it without splitting the input files, right? You gotta run probably close to a day and probably bump the core count up. Might work. Otherwise, you have to do what I said, which is you have to take the input FASTQ and break it into pieces. It doesn't really matter how. You can just, you can interleave it, just take round robin, or you can just break it into big chunks. It doesn't matter because, each read a line takes about the same amount of time per line. Anyway, the file's in a random order anyway, so whether you take an ordered or blocks out of it doesn't matter. But yeah, you partition the input FASTQ. You basically break it, you unzip it either as on disk or as a stream. You take it in blocks of 4 lines. You just make sure you take a number of lines that's a multiple of 4, because the basic FASTQ structure is read name, sequence, plus sign, quality score is 4 lines. So you just make sure you split it into blocks of 4 lines. Pick something, like 40 million lines a chunk, or 4 million lines a chunk, or whatever. Something that's maybe 10 times bigger than what we were running today, but maybe not 60 times bigger. And then that turns each input file into however many parts it has. You run BS-Bold align on all those parts, and you get a whole bunch of BAM files out, and then you merge all those BAM files together, and that's your BAM run for the thing. And that's how you can handle very large data. The job submission system is nice because you can just shove stuff into there as the job submission system will handle finding available machines, figuring out which files have been done, which files haven't, and so on. And you can just load it up and let it go, right? And things will just finish as fast as they can. So you tend to just, load it up. You may have stuff sit in the queue for days if you've really loaded up a big load of stuff to do, and it just grinds through it. Of course, the rest of the stages also possibly need separation. Everything that we talked about may need splitting into parts for very large inputs. But it's also possible up the core count, go to a day as a time limit, maybe able to squeeze it in, and that's much more convenient because you don't have to go through all the hassle of the bookkeeping of the splitting and merging.
Okay, thanks.
So Amanda, maybe we'll talk after everyone's done to figure out what's going on in your case. Okay, all right. So yeah, so we'll try to do some of the differential methylation and some of the functional. We probably won't really do a lot of comprehensive functional stuff, but we'll just give the flavor. And then I got to put together a little 10-question quiz for those of you taking it for credit, and there'll be a little homework. The homework will be designed to take 3 hours or something to do, and you'll have till next week, early next week to do that. You'll have this coming weekend to do that. I'll probably send out the quiz, either tomorrow afternoon, maybe as late as Friday morning, and so on. And you can give that to me in a day or two. That's going to be very easy 10 multiple choice questions or something. Okay, so if that's it, I'll stop the recording for today, and then if you've got questions you want to handle offline, we will do that next. Right, so stop the share, stop recording.